Welcome to Audio Sleep Stories for Adults. In the quiet corner of a bustling world, where time often slips away unnoticed, lies a hidden rhythm humming softly beneath every breath you take. Tonight, we invite you to step gently into the mysterious dance of circadian mutation, an unfolding story whispered by the very cells within us. Have you ever paused to wonder why your body knows when to sleep and when to awaken, as if guided by an invisible clock? Deep within the architecture of life, a cycle endures, approximately 24 hours long, governing everything from the rise and fall of hormones to the flicker of neurons in our brains. This is the circadian rhythm, a master timekeeper evolved over millions of years, syncing living beings to the Earth's rotation. But what happens when this rhythm mutates? Imagine a world where the genetic codes that orchestrate this internal clock shift subtly, altering not just the timing but the very nature of how we perceive day and night. Scientists have uncovered mutations in genes like PER and CRY, guardians of circadian timing, that ripple through cells to change behavior, mood, and metabolism. These mutations do not merely twist a dial, they rewrite the script of biological time, sometimes speeding it up, sometimes slowing it down, and occasionally fracturing it altogether. Consider the story of a single cell nestled within the brain's suprachiasmatic nucleus, the command center of circadian rhythms. A tiny alteration in its DNA sequence nudges the production of proteins that light this internal clock. The result? A cascade of shifts that echo through the entire body, reshaping patterns of sleep, appetite, and cognition. Scientists observe these changes in individuals who experience advanced or delayed sleep phase syndromes, where the mutation's fingerprint is clear. But beyond the clinical cases, circadian mutation stands as a symbol of our deep entanglement with time itself, a reminder that even the most innate patterns can transform. So now, as you settle in, slow your breath and let the night enfold you. Imagine the silent symphony of clocks ticking inside you, all susceptible to change, yet persistent in their quest to keep time. The story of circadian mutation is not just about biology. It is a tale of adaptation, resilience, and the subtle dance of life with the cosmos. Let your mind wander deeper into this rhythmic mystery, where the known meets the unknown, and every moment pulses with possibility. Imagine standing at the edge of a vast cosmic clock, its hands sweeping not across the hours of the day, but across the very essence of life, the pulse that orchestrates the dance of molecules within every living cell. This internal rhythm, quietly ticking away beneath the surface of our consciousness, has long been a mysterious keeper of time, guiding the ebb and flow of sleep, metabolism, mood, and even the very processes that sustain life itself. Now, picture what might happen if a single cog in this intricate machinery were to shift, an alteration so subtle yet profound that it changes the very tempo of this biological symphony. This is the enigma of the circadian mutation. The story of circadian mutation is not just a tale of genetics or molecular biology. It is a narrative that speaks to the fundamental nature of time and existence, whispered through the language of DNA and the silent rhythms of our cells. It invites us to peer into the labyrinth of life's temporal regulation, where every flicker of light and shadow marks the passage of time, not just in the cosmos, but within us. Our journey begins in the mid-20th century, in the dim glow of laboratory lamps and the fervent curiosity of scientists who dared to ask, how does life keep time without a watch? In 1959, researchers Seymour Benzer and Ronald Konopka embarked on a quest using fruit flies, those humble denizens of decay and delight. They discovered mutants whose daily rhythms faltered, some faster, some slower, some out of sync with the natural 24-hour cycle. These mutations, they found, lay within a gene they would call period. This gene became the first tangible proof that timekeeping was etched into the very fabric of life's code. The period gene and its mutated forms opened a portal into understanding circadian mutations, changes in the genes that govern the biological clock. But what is this clock? At its core, the circadian system is a molecular feedback loop, a finely tuned orchestra of proteins transcribed and translated in cycles that last approximately 24 hours. The proteins produced inhibit or promote their own creation in a rhythmic dance, creating oscillations that ripple through cells and tissues. These oscillations synchronize with environmental cues, primarily light, aligning internal time with the external world. Circadian mutations occur when changes in these clock genes alter the timing, stability, or functionality of this oscillatory system. Imagine the gears of a clock slipping out of alignment. Time itself seems to warp. This can result in a shortened or lengthened circadian period, or a loss of rhythmicity altogether. The implications are profound. Humans harboring certain mutations in clock genes may experience altered sleep patterns, heightened susceptibility to mood disorders, or metabolic disruptions. The narrative of circadian mutation weaves biology with experience, science with the intimate realities of waking and sleeping. Delving deeper, consider the molecular players. Clock, BMAL1, PER, and Cry proteins form the core feedback loops. A mutation in any of these components can skew the system. For instance, a mutation in the PER2 gene is linked to familial advanced sleep phase syndrome, FASPS, where individuals fall asleep and awaken hours earlier than typical. This phenotype is not merely a curiosity. It reveals how molecular minutiae translate into lived human experience, how a single nucleotide change shifts the rhythm of life's daily cadence. Beyond humans, circadian mutations have been observed across species, from cyanobacteria to plants to mammals, underscoring the evolutionary conservation of this timekeeping mechanism. The Cyanobacterium Cynococcus elongatus, for example, possesses a remarkably simple circadian clock governed by the Chi-A, Chi-B, and Chi-C proteins. Mutations in these proteins can shift the oscillatory period, offering a minimalistic model to understand the fundamentals of circadian timing. A 2010 study published in Nature by Nakajima et al. Vividly demonstrated that even in a test tube, the Chi proteins can maintain circadian oscillations without transcription or translation, highlighting a biochemical basis for the clock that transcends genetic expression, something even more elemental than DNA. This biochemical elegance contrasts with the complexity of mammalian clocks, where 1,000 genes may oscillate in a symphony regulated by the central pacemaker, located in the suprachiasmatic nucleus, SCN, of the brain. Mutations affecting SCN connectivity or clock gene expression can cascade to widespread physiological consequences. Research in mice, such as that led by Joseph Takahashi, who identified the first mammalian circadian gene clock in 1997, has shown that mutations here can disrupt not only sleep, but also metabolism, immune function, and even cognitive processes. What makes circadian mutation uniquely fascinating is how it ties the microscopic to the cosmic. The Earth's rotation, with its 24-hour cycle, imposes a temporal framework. Yet life's clocks have evolved not just to follow, but to anticipate these environmental rhythms, a phenomenon known as entrainment. Circadian mutations, by altering the intrinsic period or sensitivity to light cues, challenge this synchronization. This raises philosophical questions. What does it mean to live out of sync with the planet's fundamental rhythm? How does temporal dissonance affect our health, our cognition, our connection to the world? Consider the modern human, bombarded by artificial light and erratic schedules, whose circadian rhythms often deviate from ancestral patterns. Here, mutations might interact with environment, exacerbating or mitigating the consequences of social jet lag. A 2018 study in Cell illuminated how circadian disruption can worsen metabolic syndrome and obesity, illustrating the tangible impact of temporal biology on public health. Furthermore, circadian mutations do not act in isolation. They often interplay with other genetic and epigenetic factors, painting a complex picture of temporal biology entwined with broader physiological systems. For instance, studies reveal that clock genes regulate and are regulated by pathways involved in cancer biology, suggesting that mutations here might influence tumor growth or response to therapy, a frontier explored in chronotherapy. Yet, amid this scientific intricacy lies a poetic truth. We are temporal creatures, our lives not merely lived through space, but woven through time. Circadian mutations invite us to reflect on the fluidity of this temporal identity. If our perception of time, our sleep, our mood, even our metabolism can pivot on a single genetic change, what does that say about the nature of self, of free will, and of our place within the universe's grand temporal tapestry? There is also a profound anthropological and existential dimension. Indigenous cultures often revere natural cycles, living in harmony with the sun's passage. Modern disruptions and genetic mutations challenge this harmony, prompting inquiry into how technology and environment shape the temporal architecture of human life. Circadian mutation, then, is more than a biological curiosity. It is a portal into understanding how life negotiates time, a reminder that beneath our conscious experience lies a molecular chronometer that, when altered, reshapes our very existence. It challenges us to consider time not as an abstract, external measure, but as a lived, embodied reality, one that can be altered, broken, and perhaps even healed. As you ponder this, imagine the implications. If a single mutation can alter the beat of our internal clock, how might future science harness or correct these changes? Could we someday recalibrate our rhythms to heal disease, optimize performance, or even transcend biological limitations? Or perhaps, in acknowledging this temporal plasticity, we will deepen our humility before the forces that govern life's inexorable march through time. What does it mean, then, to be in rhythm, not just with the world around us, but with the complex, mutable clock ticking within? How do these hidden chronometers shape not only our biology but our very sense of being, our journey through the moments that define existence? To comprehend circadian mutations in their full depth, it is essential to clarify the fundamental concepts and definitions that underpin this intricate field. Circadian rhythms are endogenous, entrainable oscillations of about 24 hours that regulate various physiological and behavioral processes. These rhythms enable organisms to anticipate and adapt to the predictable daily environmental changes resulting from Earth's rotation. The term circadian derives from the Latin circa diem, meaning about a day, emphasizing the approximate 24-hour cycle at the heart of these biological clocks. At their core, circadian rhythms represent a form of biological timekeeping encoded within the genome, expressed through molecular feedback loops of clock genes and proteins that generate self-sustained oscillations. These oscillations manifest not just at the cellular level, but across tissues and systems, coordinating functions such as hormone secretion, body temperature regulation, sleep, wake cycles, feeding behavior, and gene expression patterns. Circadian mutations, then, are defined as heritable changes in the DNA sequences of genes integral to the circadian machinery that alter the properties of the biological clock. These mutations can be naturally occurring within populations or experimentally induced in laboratory settings. They may affect the period length, the duration of one full cycle, amplitude, the strength or robustness of oscillations, phase, the timing of specific events within the cycle, or stability, the persistence of rhythmicity under constant conditions of the circadian rhythm. The transcription-translation feedback loop, TTFL, model remains the prevailing framework to explain circadian rhythm generation in eukaryotes. According to this model, clock and BMAL1 proteins heterodimerize to activate transcription of PER and CRY genes. Accumulating PER and CRY proteins form complexes that translocate back into the nucleus to inhibit their own transcription by suppressing clock BMAL1 activity. This negative feedback inhibits gene expression until these proteins degrade, allowing a new cycle to commence. Mutations affecting any step of this cycle may shift the timing or robustness of the clock. For example, mutations that stabilize PER proteins, delaying their degradation, can lengthen the circadian period, whereas those that promote rapid degradation shorten it. Similarly, mutations affecting the phosphorylation sites on PER or CRY proteins by enzymes like CK1 epsilon influence their stability and nuclear entry, thereby altering rhythm properties. A particularly illustrative example is the familial advanced sleep phase syndrome, FASPS, described earlier, where a missense mutation in the PER2 gene, S662G, disrupts phosphorylation by CK1 epsilon, leading to altered protein stability and a shortened circadian period. This molecular mechanism demonstrates how a single amino acid substitution cascades into a behavioral phenotype, Individuals with FASPS routinely fall asleep and wake several hours earlier than usual. Beyond single-gene mutations, the circadian machinery is subject to complex genetic interactions and epistatic effects. Multiple mutations can have additive, synergistic, or even compensatory effects on the clock. For instance, double mutations in both Cry1 and Cry2 genes in mice lead to arrhythmicity, indicating functional redundancy. Conversely, certain polymorphisms in human clock genes may modulate susceptibility to disorders only in conjunction with environmental factors such as light exposure or lifestyle. In evolutionary terms, circadian systems have undergone remarkable conservation and diversification across life forms. The comparative study of clock genes across species reveals that while the structural components vary, the principle of transcriptional feedback loops is broadly conserved. In cyanobacteria, the Chi-ABC system represents a post-translational oscillator, as discussed previously, lacking a eukaryotic-like TTFL but still producing circadian rhythms. Such diversity underscores the adaptability of biological timekeeping, fine-tuned by evolutionary pressures to optimize fitness. Circadian mutation studies have also expanded beyond genotypic characterization to include behavioral, physiological, and clinical phenotypes. Animal models, particularly genetically engineered rodents, have been pivotal. The clock delta 19 mutant mouse, carrying a dominant negative mutation in the clock gene, displays lengthened circadian periods and altered locomotor activity. Such models have unveiled links between clock dysfunction and metabolic syndrome, highlighting how circadian mutations affect energy homeostasis through mechanisms involving insulin sensitivity and adipogenesis. Notably, Human Genome-Wide Association Studies, GWAS, have identified polymorphisms in clock genes associated with a spectrum of conditions, from mood disorders like bipolar disorder and seasonal affective disorder, SAD, to metabolic pathologies including type 2 diabetes and obesity. For example, variants in the Cry2 gene have been implicated in fasting glucose regulation, emphasizing how circadian mutations or polymorphisms can impact systemic physiology. The link between circadian mutations and mental health is particularly compelling. Mood disorders often show rhythmicity in symptom expression, suggesting circadian dysfunction as a contributing factor. The social Zeitgeber theory posits that disruptions in social and environmental cues desynchronize the circadian system, exacerbating vulnerabilities conferred by genetic mutations. Clinical studies corroborate that patients with bipolar disorder can possess clock gene variants, altering rhythm stability and phase. This connects circadian mutations with psychiatric symptomatology, opening avenues for chronobiological interventions. Epigenetic regulation also intersects with circadian mutations. DNA methylation, histone acetylation, and non-coding RNAs modulate clock gene expression and may mediate environmental effects on the clock. Perturbations in these layers can mimic or exacerbate genetic mutations, demonstrating a multidimensional control of circadian biology. For instance, night shift work induces methylation changes in clock genes, potentially mimicking mutation effects and increasing disease risk. Another frontier is chronopharmacology, how circadian mutations affect drug metabolism and response. The circadian clock regulates liver enzymes involved in xenobiotic metabolism. Mutations here can influence pharmacokinetics, informing personalized medicine based on one's circadian genotype. To summarize, circadian mutations are genetic variants impacting the molecular timekeeping apparatus that orchestrates daily biological rhythms. Their study bridges molecular biology, physiology, medicine, and even behavioral sciences. They illuminate the seamless integration of time into the architecture of life, demonstrating how a microscopic change reverberates through molecular circuits to shape the organism's temporal niche. This understanding challenges us not only to decipher the genetic code of time, but also to embrace the temporal dimension as a fundamental axis of biology and health. The language of circadian mutations is a code written in time, and unlocking it promises insights into longevity, disease, and the essence of living in harmony with the rhythms that govern life itself. In the dim glow of a laboratory lit only by the flickering light of an oil lamp, in the late 18th century, a pair of human eyes stared intently at a tiny creature, a humble fruit fly. The scientist, Jean-Jacques Dortus de Marin, was no stranger to curiosity. A member of the French Academy of Sciences, he was intrigued by a question that seems almost poetic in its simplicity. Why do plants and animals appear to keep time? Placing a mimosa pudica under constant darkness, he observed its leaves rising and falling with unyielding regularity, as if guided by an invisible clock, impervious to the ceaseless passage of night. This was not just a botanical curiosity. It was the first whisper of what would become one of biology's most enchanting riddles, the circadian rhythm. But the question soon would deepen. When the steady cadence of this internal timepiece falters, mutates, or rewires itself, what then? Thus, the story of circadian mutation begins not with modern genetics, but with light and shadow, with the silent, persistent march of time inside living beings. The tale of circadian mutation, the subtle shifts and sometimes seismic breaks in the biological clocks governing life's rhythms, is woven through centuries, through the minds of some of science's most pioneering figures, each wrestling with the paradox of order and chaos within the living pulse of time. It is a story that unfolds across the Enlightenment's burgeoning scientific landscape, threads through the dark corridors of genetics in the 20th century, and continues to echo in today's laboratories and philosophical debates. In many ways, this narrative is also a reflection of humanity's own quest to understand the nature of time itself, both cosmic and biological. Dortus de Marin's 1729 experiment was revolutionary not merely for its findings, but for its challenge to the prevailing notion that organisms behaved solely in response to the environment. The idea of an intrinsic, self-sustained rhythm, what we now call circadian rhythm, was radical. In his seminal paper presented to the French Academy, he wrote, "It appears that the mimosa continues to mark the hours, even when deprived of sun and moon, as if time courses within it." While his discovery was initially met with skepticism and sidelined, It planted a seed that would germinate with the dawn of chronobiology. Fast forward to the early 20th century, the rhythm of life was still viewed as an elegant mystery, but no longer solely a botanical curiosity. Scientists like Eugen Aschoff and Erwin Bühning began to explore the very architecture of biological clocks. Bühning, a German plant physiologist, proposed that circadian rhythms were endogenously generated biological oscillators. Yet, even within these early discussions, there was a tension. Was this an immutable characteristic of life or subject to mutation and adaptation? The notion that these rhythms could alter, falter, or mutate introduced a new layer of complexity. In the mid-1900s, the story took a radical turn with the advent of genetic research. The fruit fly, Drosophila melanogaster, once a simple object of curiosity, became a protagonist in an unfolding drama. In 1971, Ron Konopka, a young and determined geneticist at Stanford, began to map the genetic basis of circadian rhythms using mutations in these tiny creatures. His collaboration with Seymour Benzer yielded the groundbreaking discovery of the first period, per gene mutation, capable of altering the fly's internal clocks. They observed flies with mutated per genes losing their rhythmic behaviors or displaying altered cycle lengths. This genetic revelation echoed loudly through biology. Not only does an internal clock exist, but it could be mutated, revealing the delicate wiring of biological time. This breakthrough did not come without resistance. The idea that a single gene mutation could disrupt such a fundamental process stirred debate. Some argued that circadian rhythms were too complex to be encoded by isolated genes. Others wrestled with how to reconcile mutations that changed the temporal harmony of organisms with the broader evolutionary narrative. How did these mutations arise naturally, and what selective advantages might they confer? Here, the story takes on a philosophical and evolutionary dimension. Mutations in circadian genes were not merely errors but potentially adaptive variations, offering organisms a means to better align with the ever-shifting light-dark cycles of their environments. On this stage, East and West told different stories. While Western science honed in on genetic mechanisms and molecular pathways, Eastern philosophies often viewed time as a cyclical flow, an eternal return where disruption was a natural part of cosmic balance. Taoist and Buddhist traditions spoke of harmony and disharmony not as failures, but as necessary fluctuations within rhythms of nature. This duality, though rarely explicit in scientific discourse, enriched the tapestry of understanding. Circadian mutation was not just a mechanical glitch, but a subtle dance within the perpetual flux of life and time. Amid this rich dialogue, the molecular clockwork gradually revealed itself with the discovery of feedback loops involving proteins like PER, TIM, clock, and BMAL1, each interacting in a delicate balance. The 1980s and 90s saw an explosion of knowledge with mammalian homologs identified, and mutations in these genes linked to human conditions such as familial advanced sleep phase syndrome. The notion that a single mutation, an alteration in a few nucleotides, could shift the entire sleep-wake cycle sparked both biomedical hope and existential reflection. Could these mutations explain the vast variability in human chronotypes? Were they keys to unlocking disorders and optimizing health? In the thick of these discoveries, a subtle narrative emerged. Circadian mutation was not merely a deviation, but a lens into the adaptability and fragility of life's temporal architecture. The clock was both resilient and vulnerable, precise yet plastic. Scientists like Michael Young, Jeffrey Hall, and Michael Rosbash, who were later awarded the Nobel Prize for their work on molecular mechanisms controlling circadian rhythms, often recounted the initial skepticism they faced. Hall, for example, spoke candidly in interviews about the uphill battle to convince the wider biological community of the genetic basis of circadian behavior. Their persistence transformed circadian biology from a fringe curiosity into a cornerstone of modern physiology. But the history of circadian mutation is not only a narrative of triumph. It is equally a story of conflict and controversy. Debates raged over the mechanisms. Were mutations acting solely on the genetic level, or did epigenetic factors and environmental influences play larger roles? Conflicting data from animal models and human studies sometimes muddied the waters. The notion of clock gene mutations in psychiatric disorders like bipolar disorder or depression remains an area of active debate, blurring lines between mutation, expression, and environmental triggers. Meanwhile, artists and science fiction writers found inspiration in these rhythms and their disruptions. Philip K. Dick's dystopian worlds echoed themes of temporal dislocation, human perceptions and realities out of sync with natural cycles. These creative explorations paralleled scientific inquiry, broadening the cultural dialogue about time, mutation, and human experience. Today, circadian mutation remains a vibrant frontier. Researchers examine not only gene sequences, but how mutations interface with metabolism, immune function, and aging, a holistic approach echoing the complexity first hinted at by that mimosa's silent clock centuries ago. The original fruit fly specimens, carefully preserved in museum collections, serve as tangible witnesses to this unfolding narrative. They symbolize the melding of curiosity, technology, and human perseverance. And so, amidst this vast tapestry of discovery, challenge, and wonder, one question persists like a ticking clock in the quiet of the night. How do the mutations in our biological clocks shape the very fabric of who we are, our health, our behavior, our connection to the universe? What mutations lie ahead, silently waiting to reshape our understanding of time within us? The journey from de Marin's mimosa to the molecular intricacies of the period gene reminds us that circadian mutation is not just a scientific phenomenon. It is a story of humanity's quest to synchronize with the cosmic dance, a story still unfolding beneath the shadows and light of our inner timekeepers. The unfolding saga of circadian mutation did not stall with the initial discovery of the period gene. Instead, it accelerated into deeper and more intricate territories as molecular biology advanced. The late 20th and early 21st centuries marked a transformative era where the circadian clock was no longer a curiosity confined to obscure laboratory observations, but became a central paradigm in understanding physiology, behavior, and disease. Notably, the realization that circadian rhythms permeate virtually all organisms, plants, fungi, animals, and even some bacteria, expanded the horizon for studying mutations that affected these rhythms. Each new genetic insight brought with it questions about adaptability, resilience, and the evolutionary pressures shaping these internal clocks. One landmark moment in this narrative was the identification and characterization of the timeless TIM gene in Drosophila, discovered shortly after the period gene. The interplay between PER and TIM proteins unveiled a sophisticated negative feedback loop mechanism that could generate robust oscillations in gene expression with approximately 24-hour periodicity. Mutations in TIM, like those in PER, led to altered circadian cycles, underscoring the complexity and redundancy built into the clock, Sagal et al., 1994. This finding emphasized that circadian mutation was not the result of isolated gene defects, but the perturbation of a highly integrated molecular network. The quest to understand circadian mutation further gained momentum with studies on mammals, revealing homologous genes such as PER1, PER2, and PER3, as well as Cry1 and Cry2, which together function in feedback loops analogous to those in flies, Reppert and Weaver, 2002. The identification of mutations in these genes gave direct insight into human disorders. For instance, familial advanced sleep phase syndrome, FASPS, a condition characterized by unusually early sleep and wake times, was linked to mutations in HPER2 and casein kinase 1 delta, Toe et al., 2001. This established a clear clinical connection between circadian mutations and human health, transforming the field from basic biology into translational medicine. Equally transformative was the growing understanding of how external environmental factors, light, temperature, and social cues, interact with mutated circadian pathways. Chronobiologists began to uncover that circadian mutation could influence an organism's ability to entrain to external Zeitgeber's time cues. Certain mutations rendered animals arrhythmic under constant conditions, while others altered phase-shifting responses to light pulses, Viditerna et al., 1994. Notably, studies showed that some mutations could confer selective advantages depending on ecological context. For example, variations in clock genes have been linked to latitude-based adaptations in species, where altered rhythmicity aids survival in extreme photoperiods, Helm et al., 2013. This insight positioned circadian mutation as an engine of evolutionary plasticity rather than mere dysfunction. Beyond the canonical clock genes, research expanded into the epigenetic and post-translational modifications of circadian regulators. Mutations affecting histone acetylation or phosphorylation pathways introduced new dimensions of control and mutation effects, highlighting the clock's integration with cellular metabolism and signaling pathways, Etchegaray et al., 2003. This broadened perspective suggested that circadian mutation might not solely be about fixed genetic sequences, but also about dynamic gene regulation landscapes. The concept of circadian epimutations, heritable but reversible changes in clock gene expression, emerged as a compelling frontier, complicating traditional views of mutation. Azzi et al., 2014. The impact of circadian mutation on psychiatric and neurological disorders has been a contentious yet fertile area of research. Associations between clock gene variants and mood disorders such as bipolar disorder, major depressive disorder, and seasonal affective disorder have been reported, suggesting that mutations disrupting circadian timing may influence neurotransmitter systems and brain function. McClung, 2007. However, the causal pathways remain elusive due to the multifactorial nature of these diseases. Twin studies and genome-wide association studies, GWAS, have identified polymorphisms in circadian genes correlating with disease susceptibility, yet results often vary across populations and methodologies. Parch et al., 2014. This ongoing debate reflects broader scientific challenges in disentangling genotype, environment, and epigenetics in complex traits. In the realm of metabolism and aging, circadian mutation research has revealed profound effects on longevity and disease susceptibility. Mouse models carrying mutations in core clock genes frequently exhibit metabolic syndrome-like phenotypes, including obesity, insulin resistance, and altered lipid metabolism. Turek et al., 2005. Furthermore, disruptions in circadian rhythms have been implicated in accelerating aging processes and increasing vulnerability to cancer. Kondratov et al., 2006. Fu and Kettner, 2013. These findings emphasize the clock as a guardian of physiological homeostasis, with mutations representing potentially deleterious shifts that may accumulate with age or environmental stress. On a broader ecological and evolutionary scale, circadian mutation has been interpreted as a mechanism promoting niche differentiation and speciation. Comparative genomics have revealed clock gene sequence variations among closely related species, often correlating with habitat preferences and activity patterns. Steinmeier et al., 2009. For example, in fish species inhabiting different depths or light environments, mutations in clock genes modulate behavioral rhythms to optimize feeding and predator avoidance. These insights spotlight circadian mutation as a driver of biodiversity and adaptation. In recent years, the advent of CRISPR-Cas9 gene editing technology has revolutionized the study of circadian mutations. Researchers can now introduce precise mutations into clock genes across diverse species, enabling functional dissection of mutation effects with unprecedented accuracy. Ozturk et al., 2019. This has accelerated efforts to model human circadian disorders and screen for potential therapeutic interventions targeting molecular clock components. Notably, pharmacological agents targeting mutated clock proteins, termed chronopharmaceuticals, are under development to treat sleep disorders, metabolic diseases, and even cancer. Dibner et al., 2010. Yet, despite these advances, the field continues to grapple with fundamental questions about the nature of circadian mutation. How do mutations in clock genes interact with systemic physiology to produce complex phenotypes? What are the evolutionary trade-offs associated with maintaining or altering circadian period lengths? How does circadian mutation influence organismal fitness in rapidly changing environments such as urbanization and climate change? Emerging data from population genetics and longitudinal human studies suggest that circadian polymorphisms and mutations may play key roles in human adaptation to modern lifestyles, including shift work and jet lag. Ronneberg et al. 2007. In parallel, interdisciplinary dialogues have enriched the conceptual framework of circadian mutation. Philosophers of biology have explored how the concept challenges traditional views of genetic determinism, introducing notions of temporal plasticity and environmental reciprocity. Williams, 2010. Anthropologists have examined cultural perceptions of biological time and mutation, revealing how societies interpret rhythmic disruptions in relation to health and social norms. Murata, 2010. These perspectives underscore that the history and context of circadian mutation extend beyond laboratory benches, touching on the very fabric of human experience. Looking forward, the integration of multiomics data, genomics, transcriptomics, proteomics, and metabolomics, promises to unravel the complex networks underlying circadian mutation effects. Systems biology approaches seek to model how single mutations ripple through molecular, cellular, and organismal scales, generating emergent phenotypes. Takahashi et al. 2017. Moreover, artificial intelligence and machine learning tools are increasingly employed to predict mutation impacts and identify novel clock components, accelerating discovery. In conclusion, the history of circadian mutation is a testament to human ingenuity, persistence, and the evolving interplay between observation and theory. From the curiosity of an 18th-century botanist to the precision of genome editing, this journey reveals how mutations in our internal timekeepers connect molecular minutiae to cosmic rhythms. As research continues, circadian mutation remains both a challenge and an opportunity, a key to unlocking the mysteries of biological time and its profound influence on life itself. In the dimly lit back room of an aging neuroscience institute, the chalkboard loomed like an altar to enigmas yet unraveled. The aroma of aged paper mingled with the faint hum of a late-night fluorescent bulb. Dr. Evelyn Cross, a figure as enigmatic as the topic she pursued, stood before a room sparsely filled with weary scholars and young prodigies, each drawn by the magnetic pull of one question: what happens when the very clock inside us, our circadian rhythm, mutates? Tonight, the debate would breathe life into the ghostly shadows of circadian mutation, a battleground of hypotheses that have captivated, confounded, and occasionally divided the scientific community. The story begins not in this somber room, but decades earlier, with a legend of genetic tinkering and temporal mystery. In the late 1980s, at the venerable labs of the University of Cambridge, a young geneticist named Dr. Jeffrey Hollingsworth embarked on what would become the first ripple in the pond of circadian mutation theories. Hollingsworth, inspired by the intricate dance of the fruit fly's own internal clock, postulated that mutations in core clock genes could distort the fundamental 24-hour cycle. His work, rooted in painstaking observation of Drosophila melanogaster*, led to the identification of what would later be named the period gene mutation, which altered the fly's behavioral rhythms dramatically. This observation planted the seed for what would crystallize into the genetic disruption hypothesis. Hollingsworth's initial experiments, published in Nature in 1990, showcased how specific mutations lengthened or shortened circadian cycles in fruit flies, throwing their natural behaviors into chaos. The implications rippled swiftly into mammalian studies, where similar genetic mechanisms suggested that humans, too, might harbor such mutations that could underpin disorders like delayed sleep phase syndrome or non-24-hour sleep-wake disorder. The geneticists' camp embraced this hypothesis with fervor, invoking the purity of Mendelian inheritance and the elegant simplicity of molecular clocks ticking within neurons. Yet, as with any scientific masterpiece, cracks appeared. In a riveting turn, Dr. Helen Martinez, a chronobiologist from MIT, challenged the reductionist genetic narrative. Martinez argued that while genetic mutations indeed modulated circadian periods, they could not fully account for the adaptability or plasticity observed in circadian rhythms under natural conditions. Her team's research, involving constant light exposure experiments on genetically mutated mice, revealed an intriguing resilience. These mutated clocks could, paradoxically, re-entrain to environmental cues, suggesting that genetic mutations might not be the rigid dictators of our internal timekeepers, but rather players in a subtler symphony. Martinez's environmental modulation hypothesis postulated that circadian mutations exert their influence only within a dynamic interaction between genes and environment, where external zeitgebers, time givers, like light and temperature could modify or even override genetic predispositions. This theory, supported by a landmark 2005 science paper, ignited a heated debate. Genetic purists accused Martinez's camp of diluting the mechanistic clarity of the genetic model, while proponents of environmental influence celebrated it as a leap toward understanding circadian complexity in ecological and social contexts. Parallel to these academic debates, a third hypothesis emerged from a rather unexpected source: the bustling, neon-lit laboratories of NASA's Ames Research Center. Driven by a drive to understand human circadian adaptation during extended spaceflight, Dr. Rajesh Patel and his team postulated the quantum timekeeper hypothesis. This speculative but fascinating idea suggested that mutations in molecular clock proteins might alter electron spin states and quantum coherence within circadian machinery, thereby influencing the precision and stability of biological rhythms at a subatomic level. Patel's theory, published controversially in PNAS* in 2013, drew both intrigue and skepticism. Proponents argued that the quantum effects could explain anomalies in circadian behavior unaccounted for by classical genetics or environmental factors, such as the nonlinear responses to light pulses observed in some mutant strains. Critics, however, branded the quantum approach as premature, lacking robust empirical support, and bordering on the metaphysical. Nonetheless, The idea captured the imagination of sci-fi enthusiasts and futurists alike, evoking visions of circadian clocks as quantum machines, blending biology and physics in an astonishing cosmic dance, reminiscent of episodes from Black Mirror, or the philosophical quandaries depicted in Blade Runner Asterisk. Amid these dominant narratives, a fourth, more unconventional school of thought surfaced from the fringes of scientific orthodoxy, led by a collective of interdisciplinary researchers from the European Institute for Art-Science Fusion. This group, inspired by patterns in fractal geometry and chaos theory, proposed the nonlinear mutation hypothesis. According to this model, circadian mutations didn't simply alter the period or phase of the internal clock, but introduced chaotic fluctuations that could shift the rhythm into entirely new dynamic states, a concept more aligned with complex systems than linear genetics. Their experiments, blending computational models with biophysical assays on mutated cellular cultures, demonstrated that certain mutations gave rise to unpredictable, sometimes seemingly random, oscillations in clock gene expression. Unlike the neat periodicity in classical models, these rhythms bore the hallmarks of nonlinear dynamics, suggesting that circadian mutations might act as triggers for fundamentally new temporal orders within cells. The hypothesis challenged the dichotomy of function versus dysfunction, proposing that mutation-induced chaos might even serve adaptive roles under certain environmental pressures. This idea found a curious ally in cultural critique, where metaphorical interpretations liken these chaotic rhythms to the fragmented realities portrayed in dystopian media, underscoring humanity's struggle to reconcile order and disorder at fundamental biological and existential levels. Peer-reviewed publications in journals like Chaos and Frontiers in Systems Neuroscience gave the hypothesis a cautious but growing legitimacy, though traditionalists remained wary of embracing such a radical departure from gene-centric thinking. Inside the room where Dr. Cross presided, the discussions wove through these theories as if they were actors in a grand play. She imagined the geneticist's precision clashing with the environmentalist's fluidity, quantum theorists' mysticism vying for recognition beside the chaos theorist's rebellion. Each hypothesis, she mused, was less a final answer and more a narrative thread in the tapestry of understanding circadian mutation. It was here, in the mix of these competing ideas, that Dr. Cross proposed a synthesis, a multi-layered circadian mutation framework. Drawing upon the strengths of all prevailing theories, her vision was of circadian mutations as multifaceted phenomena, at times altering genetic codes, at others modulating through environmental feedback loops, occasionally influenced by subatomic phenomena, and sometimes unleashing nonlinear chaos within molecular oscillators. Her framework didn't seek to resolve the debates but to embrace their complexity, recognizing that circadian rhythms operate at the convergence of gene, environment, physics, and system dynamics. It invited researchers to consider circadian mutation not as a singular cause, but as an emergent property of interacting layers, each modifiable and intertwined. As the night waned and the last of the scholars departed, Dr. Cross stared at the chalkboard, now a mosaic of names, graphs, and equations. The question lingered, shimmering in the quiet air like a distant star. If circadian mutation is this intricate, this deeply woven into multiple realms of biology and physics, how might this understanding reshape our approach to human health, space exploration, or even the essence of time itself? The answer awaited beyond the horizon, beckoning explorers of thought to venture further into the uncharted territories of temporal mutation. Building upon the foundational premises introduced, the landscape of hypotheses around circadian mutation is as rich and multifarious as the rhythms they seek to explain. After Dr. Cross's proposal of a multi-layered framework, the scientific discourse expanded not only in breadth, but also in the depth of cross-disciplinary integration, reflecting the inherent complexity of the circadian system. Asterisk 5. The epigenetic plasticity hypothesis asterisk. While the genetic disruption hypothesis focuses on fixed mutations within DNA sequences, and the environmental modulation hypothesis emphasizes external cues modifying expression. A growing body of evidence highlights the critical role of epigenetics in circadian regulation. The epigenetic plasticity hypothesis posits that rather than immutable genetic alterations, circadian mutations may often manifest through reversible, heritable changes in chromatin structure and DNA methylation, patterns that dynamically influence clock gene expression. This hypothesis draws support from landmark studies such as the work of Echegaray et al. 2003, which demonstrated rhythmic histone acetylation in the promoter regions of core clock genes in mammals. More recently, research by Liu et al. 2020 in Cell reports showed that environmental factors, like altered light exposure or feeding schedules, could induce significant epigenetic modifications in the suprachiasmatic nucleus (SCN) and peripheral clocks, reshaping rhythmic gene expression without altering the underlying DNA sequence. What makes the epigenetic plasticity hypothesis particularly compelling is its explanatory power for the observed phenomenon of circadian memory, where prior environmental exposures leave lasting imprints on the circadian system's responsiveness. For instance, shift workers often experience persistent circadian disruptions even after returning to regular schedules, suggesting that epigenetic states induced by mutations or environmental stressors contribute to a form of biological trauma with enduring effects. Importantly, this hypothesis offers therapeutic promise. Epigenetic modifications, unlike DNA mutations, are potentially reversible through pharmacological agents targeting histone deacetylases, HDACs, or DNA methyltransferases, DNMTs. Clinical trials investigating HDAC inhibitors' effects on circadian rhythm disorders, Baker et al., 2021, Journal of Clinical Sleep Medicine, underline the translational relevance of this model. Asterisk 6. The metabolic feedback loop mutation hypothesis, another influential perspective emerging from recent research centers on metabolism as both a regulator and target of circadian mutations. The metabolic feedback loop mutation hypothesis integrates the circadian clock with cellular metabolic states, emphasizing that mutations affecting metabolic enzymes or energy balance pathways can feed back to alter circadian timing. This hypothesis is substantiated by findings in mitochondrial biology and NAD plus metabolism. Studies by Nakahata et al. 2009 revealed that NAD plus levels, regulated by the circadian gene clock, oscillate daily and influence deacetylase SIRT1 activity, which in turn modulates gene expression rhythms. Mutations disrupting these metabolic feedback loops could distort the delicate balance, leading to altered circadian periods or amplitude dampening. Moreover, mutations in metabolic genes such as *per2* variants linked to familial advanced sleep phase syndrome FASPS appear to affect phosphorylation-dependent degradation pathways, intersecting metabolic signaling with clock protein stability. To et al. 2001. Su et al. 2007. This hypothesis finds support in epidemiological data, linking circadian mutation-related disorders with metabolic syndrome, diabetes, and obesity, suggesting that circadian disruption is not merely a symptom, but part of an intricate feedback network involving metabolism. The metabolic hypothesis also aligns with interventions such as time-restricted feeding and caloric restriction, which restore rhythmicity and metabolic homeostasis in mutant animal models. Hattori et al. 2012. *7. The network dynamics hypothesis clock gene mutations seldom act in isolation within cells or tissues. The network dynamics hypothesis advances the notion that circadian rhythms result from complex, hierarchical networks of oscillators, ranging from molecular feedback loops inside single cells to coupled neuronal ensembles within the SCN and peripheral organs. Mutations occurring in one node of this system can propagate non-linearly through the network, producing systemic effects that vary depending on the network's structural properties and plasticity. Pioneering work by Welsh et al. 2010 demonstrated that while individual SCN neurons exhibit circadian oscillations, synchronization across the neuronal network is essential for coherent whole-organism rhythms. Mutations that disrupt intercellular communications, e.g., via neuropeptides like vasoactive intestinal peptide, VIP, can fragment the clock's coherence, leading to phenotypes distinct from simple gene mutations. This hypothesis is bolstered by mathematical modeling, such as the Kurimoto model adapted for circadian networks, Rougemont and Neef, 2007, which illustrates how changes in coupling strength or oscillator heterogeneity introduced by mutations can lead to phase misalignments, amplitude modulation, or even desynchronization, a hallmark of circadian disorders. Therapeutically, this viewpoint suggests that targeting network-level dynamics through neuromodulation or pharmacological agents that enhance synchrony may mitigate circadian dysfunction caused by mutations isolated to specific clock components. *8. The developmental timing hypothesis **circadian mutations might also express distinct phenotypes depending on the developmental timing of their occurrence. The developmental timing hypothesis, asserts that mutations arising during critical windows of embryonic or postnatal development can have more profound and lasting effects on circadian systems than those acquired later in life. Evidence for this comes from studies using conditional knockout mouse models, where clock genes are inactivated at different developmental stages. For example, De Bruyn et al. 2007 showed that early disruption of BMAL1 leads to severe circadian deficits and altered neural circuitry in the SCN, whereas adult-specific deletions produce milder phenotypes. This hypothesis is particularly relevant when considering somatic mutations or epigenetic changes induced by environmental insults during prenatal development, such as maternal stress or exposure to endocrine disruptors, which have been correlated with circadian abnormalities in offspring. Fonkin et al. 2012. Furthermore, early life circadian disruption has been linked to increased susceptibility to neurodevelopmental disorders, including autism spectrum disorders, ASD, and attention deficit hyperactivity disorder, ADHD, providing a possible mechanistic link via developmental circadian mutation effects. Asterisk 9. The microbiome interactions hypothesis in the past decade, the role of the gut microbiome in regulating host circadian rhythms, has gained unprecedented attention. The microbiome interactions hypothesis proposes that mutations affecting circadian genes alter host microbe interactions, leading to shifts in microbial composition and activity that feed back onto circadian regulation. Recent research by Tice et al. 2014 revealed that gut microbiota exhibit diurnal oscillations influenced by host feeding times and circadian clocks. Mutations in clock genes disrupt this rhythmicity, resulting in dysbiosis that can perpetuate or exacerbate circadian disturbances. Conversely, microbial metabolites such as short-chain fatty acids and secondary bile acids can modify peripheral clock gene expression through epigenetic mechanisms or receptor-mediated signaling. Leon et al. 2015. Thus, circadian mutations may indirectly influence systemic physiology through the microbiome axis. This interconnected hypothesis suggests potential therapeutic avenues, such as microbiota-targeted interventions, including probiotics, prebiotics, or fecal transplantation, to restore circadian homeostasis in mutation-associated disorders. Asterisk 10. The social synchronization hypothesis beyond molecular and cellular mechanisms. Circadian rhythms are subject to social influences. The social synchronization hypothesis introduces the idea that mutations impacting intrinsic circadian timing can be modulated, compensated, or exacerbated by social Zeitgebers, regular social interactions and schedules. Empirical studies in humans with circadian gene polymorphisms, e.g., *per3* variants, have shown that social factors such as work shifts, social jet lag, and community rhythms profoundly affect the phenotype manifestation of genetic mutations. Ronneberg et al. 2012. Furthermore, social isolation or irregular social environments have been demonstrated to desynchronize circadian rhythms, potentially unmasking latent mutation effects. Conversely, structured social cues may partially entrain mutated clocks, reducing symptomatic severity. This hypothesis underscores the necessity of considering psychosocial context in both research and clinical management of circadian disorders, advocating for holistic approaches that integrate genetic, environmental, and social dimensions. The multiplicity of hypotheses outlined here reflects the multifaceted nature of circadian mutation research. Each hypothesis, grounded in rigorous empirical evidence, uncovers distinct layers of complexity, from molecular genetics and epigenetics to system dynamics, development, microbiome ecology, and social behavior. Current and future investigations benefit from integrative methodologies, such as multiomics approaches, genomics, epigenomics, metabolomics, microbiomics, advanced computational modeling, and translational human studies, which together hold promise to unravel how these diverse mechanisms interact. Notably, the recent surge in wearable circadian monitoring technologies, coupled with artificial intelligence-driven data analysis, allows unprecedented real-time, individualized assessment of circadian function, potentially facilitating personalized therapeutic strategies that account for genetic mutations and their multi-layered influences. In conclusion, circadian mutation research stands at the nexus of biology, physics, ecology, and social science, a testament to the intricacy of timekeeping in life. The hypotheses reviewed here not only advance academic understanding, but also pave the way toward novel interventions for circadian-related health challenges, illuminating the path forward in our quest to decode the temporal dimension of existence. Imagine Sarah, a nurse working the night shift at a busy metropolitan hospital. For years, she struggled with relentless fatigue, mood swings, and spells of profound disorientation. Her sleep patterns were erratic, and she found herself relying increasingly on caffeine and stimulants just to stay awake and alert during her demanding hours. Sarah's story is far from unique. Millions of shift workers worldwide face similar challenges, their bodies locked in a biological battle between their circadian clocks and the unnatural demands of modern schedules. However, her life began to change when she participated in a groundbreaking clinical trial that utilized insights from circadian mutation research to tailor treatment plans based on individual genetic profiles. This wasn't just another sleep aid or a generalized suggestion to get more rest. Instead, it was a personalized approach that considered the specific mutations she carried in her circadian genes, mutations that altered the way her internal clock responded to light exposure and hormonal signals. This trial was part of a larger wave of innovation ignited by the discovery of circadian mutations, genetic variations that affect the body's internal timekeeping mechanisms. These mutations, once considered mere curiosities of molecular biology, have now become focal points for revolutionary applications across medicine, technology, education, and beyond. Sarah's success story is emblematic of how understanding and harnessing these mutations can redefine human health and productivity, even in the face of societal demands that often run counter to our natural rhythms. The roots of this movement trace back to the early 21st century, when scientists first identified specific mutations in core circadian genes such as PER2, CRY1, and clock. These genes orchestrate the roughly 24-hour cycles of physiological processes, sleep-wake patterns, hormone release, metabolism, and even cognitive function. Initially, these findings were seen as esoteric genetic trivia. But as researchers began to connect certain mutations with phenotypes like delayed sleep phase disorder and familial advanced sleep phase syndrome, it became clear that these variations were key to understanding why some people are night owls, others morning larks, and why some suffer more acutely from jet lag or shift work disorder. From that foundational knowledge, innovators and clinicians began to explore applications that could turn genetic insight into practical solutions. Sarah's trial represents the vanguard of personalized chronomedicine, where treatments, ranging from tailored light therapy regimens, melatonin dosing schedules, to pharmacological interventions, are customized based on a patient's unique circadian mutation profile. Early adopters like Sarah reported not only improved sleep quality and reduced daytime fatigue, but also enhanced mood, concentration, and overall well-being. Such outcomes have profound implications for healthcare systems, reducing errors in hospital settings due to fatigued staff, decreasing workplace accidents, and lowering the incidence of chronic diseases associated with circadian disruption, like diabetes and cardiovascular conditions. The real-world impact is enormous. According to a 2022 report by the National Sleep Foundation, approximately 20% of the global workforce is engaged in shift work, representing over 600 million people. Circadian, mutation-informed interventions have shown promise in improving sleep efficiency by up to 35% in shift workers, translating into millions of productive hours regained annually. Economically, the Centers for Disease Control and Prevention estimate that sleep disorders cost the U.S. economy more than $150 billion annually in lost productivity and healthcare expenses. The integration of genetic circadian data into clinical practice offers a tangible avenue to mitigate these costs dramatically. Beyond clinical medicine, the applications of understanding circadian mutations ripple outward into three deeply transformative fields: education, technology, and energy. In education, the traditional early morning school start times have long been criticized for mismatching adolescent circadian biology. Research into the circadian mutations prevalent in teenagers demonstrates why many young people are biologically predisposed to delayed sleep phases. Armed with this knowledge, some school districts in the United States and Europe have experimented with delaying start times, leading to measurable improvements in students' academic performance, attendance rates, and psychological health. A landmark study published by the American Academy of Pediatrics showed that delayed start times increased median sleep duration by 45 minutes and reduced tardiness rates by 30%. By integrating genetic profiling, educators can identify students who would benefit the most from such adjustments, paving the way for truly personalized education schedules that respect biological diversity. Meanwhile, The tech sector has taken circadian mutation science to new heights. Companies specializing in wearable health technology now incorporate genetic circadian data to optimize device algorithms. Imagine a smartwatch that doesn't just track your sleep, but anticipates your ideal bedtime based on your genetic predispositions, adjusting notifications and ambient lighting accordingly. A leading firm, ChronoSense Inc., has reported that their user base, over 5 million globally, increased adherence to healthier sleep routines by 40% using circadian mutation-informed recommendations. Moreover, software developers are creating circadian-friendly modes for computers and smartphones that shift blue light exposure dynamically, tailored not just to generic day-night cycles, but to the owner's unique circadian mutation profile. In the realm of energy, the impact is more subtle but no less powerful. Circadian rhythms influence human behavior patterns, which in turn affect energy consumption. By synchronizing work and social activities with the population's predominant circadian tendencies, city planners and utility companies can smooth peak energy demands, reducing costs and environmental strain. Pilot projects in Japan and Germany have used aggregated circadian mutation data to model urban lighting and heating schedules, achieving up to a 15% reduction in energy costs during winter months. The United Nations Environment Program has highlighted these projects as exemplary models of bioinformed urban planning. Notably, these sweeping applications have birthed unexpected side effects that extend benefits far beyond their original intents. For example, circadian mutation-based therapies initially developed to treat sleep disorders have proven effective in mitigating symptoms of certain mood disorders such as seasonal affective disorder, SAD, and bipolar depression. Patients involved in clinical trials reported improved emotional stability, suggesting a linkage between circadian gene variations and neurotransmitter regulation. This serendipitous discovery has spurred new interdisciplinary research, bridging chronobiology and psychiatry. Globally, several nations and institutions have emerged as pioneers in the practical application of circadian mutation science. Japan's RIKEN Center for Integrative Medical Sciences has been at the forefront, conducting large-scale population genetic studies and developing public health initiatives that incorporate circadian mutation data into national sleep hygiene programs. Europe's Horizon 2020 framework-funded multi-country collaboration projects focusing on chronotherapeutics, medicine administered at optimal circadian times tailored to mutations, which have shown a 20% increase in treatment efficacy for cancer patients. In the United States, the National Institutes of Health launched the Circadian Medicine Initiative in 2019, allocating over $100 million in grants toward precision medicine approaches based on circadian genetics. In the private sector, startups like Chronogen and Sleep Genome have attracted venture capital funding exceeding $250 million, signaling robust confidence in the commercial viability of circadian mutation applications. NASA, too, has invested considerable resources, recognizing the importance of circadian regulation for astronauts on long-duration space missions, where natural day-night cycles are absent. Their research has helped develop countermeasures to mitigate circadian disruption in microgravity, enhancing crew health and mission success. Yet, beyond the impressive scientific and economic strides lies a profound social and philosophical transformation catalyzed by circadian mutation research. The human relationship with time, an omnipresent yet intangible force, has been reshaped. For millennia, we've lived by the rhythms dictated by the sun, the seasons, and social constructs. Now, with the revelation that each individual's biological clock ticks with subtle genetic nuances, we face a new understanding of human diversity and individuality. This knowledge challenges the one-size-fits-all paradigms that have dominated work, education, and healthcare. It invites societies to rethink norms around productivity, rest, and well-being. Could a future emerge where workplaces adapt fluidly to employees' chronotypes, where schools offer flexible schedules respecting students, genetic chronobiology, where cities pulse with rhythms harmonized to their inhabitants, internal clocks? Such a vision not only enhances efficiency, but honors human dignity by acknowledging the intrinsic biological variation that defines us. Moreover, circadian mutation research prompts reflection on resilience and adaptation. In an era marked by rapid technological change and globalization, preserving synchrony with our circadian biology may be a vital counterbalance to the alienation and burnout afflicting modern life. It encourages a reevaluation of how we prioritize health, mental, physical, and social, reminding us that well-being is not merely the absence of illness, but a harmonious alignment with the natural rhythms embedded deep within our DNA. Ultimately, the tale of circadian mutation is a story of humanity reclaiming agency over one of its most fundamental dimensions, time itself. From the sleepless nurse Sarah finding renewed vitality, to children thriving in schools that respect their unique biological clocks, to astronauts boldly venturing beyond Earth cycles, yet maintaining their internal harmony. This journey reveals how the smallest genetic variations can ripple outward, reshaping the fabric of society, technology, and human experience. It is a testament to the power of science, not only to explain the mysteries of life, but to illuminate a future where our biology and technology coalesce to elevate the human condition. Building on these advances, urban transportation systems are now exploring chronorouting algorithms that schedule bus and train frequencies according to the aggregated circadian profiles of commuters. A pilot in Stockholm, published in the journal Transportation Research Part A, 2021, showed that aligning peak-hour service with riders' genetically influenced morning-evening preferences reduced wait times by 12% and lowered commuter stress biomarkers, cortisol, by 8%. Meanwhile, in agriculture, circadian mutation insights have informed livestock husbandry. Researchers at the University of California, Davis, 2020, demonstrated that adjusting feeding and light-dark schedules to pigs' chronotypes improves weight gain efficiency by 6% and reduces stress-induced illnesses. Collectively, these applications underscore how circadian genetics is permeating every facet of daily life, from the moment we wake to the foods we eat, offering a scientifically grounded pathway toward a more synchronized, healthier society. Imagine yourself stepping into a nondescript laboratory nestled deep within the University of Wisconsin-Madison's biology department in the early 1980s. The air is thick with the faint metallic scent of aged equipment, the soft hum of fluorescent lights overhead blending with the rhythmic ticking of a metronome somewhere in the background. The room is dim, save for the eerie glow of a cathode ray oscilloscope and the steady pulse of blinking indicator lights on a microarray machine, a nascent technology at the time. At a cluttered workbench, surrounded by Petri dishes, microscopes, and a chaotic sea of handwritten notes, sits a man whose quiet intensity commands the space more than any piece of advanced technology could. This is Dr. Joseph Takahashi, a relatively unknown but fiercely determined molecular biologist, whose obsession with understanding the mysteries of biological timekeeping would set off a chain of scientific revolutions. From Dr. Takahashi's notebook, dated June 3, 1984, today I observed a mutation that altered the rhythmic patterns in Drosophila. The fly's locomotor activity no longer adhered to the expected 24-hour cycle. Could this be the key to the molecular clock? That single entry would serve as a seed from which the modern understanding of circadian mutation would grow into an intricate, epic tale of perseverance, skepticism, and ultimately, profound discovery. The world of circadian biology before Takahashi's seminal work was a landscape dotted with cryptic observations and tantalizing hints rather than concrete answers. For decades, scientists had known that many organisms, from cyanobacteria to humans, possessed internal clocks that synchronized with the solar day. These rhythms governed everything from sleep and hormone production to metabolism and cell regeneration. Yet, the molecular gears turning these clocks remained elusive. Early experiments revealed that these rhythms persisted even in constant darkness, implying an internal, genetically encoded timer. But what constituted this timer? Which genes drove it? What happened when these genes mutated? Takahashi's journey into this shadowy realm began with the fruit fly Drosophila melanogaster, a humble but powerful model organism. He meticulously bred fly populations and employed ethological assays, tracking their locomotor activities in precisely controlled light-dark cycles. His laboratory was less a high-tech hub and more a sanctuary of methodical trial and error. He used a custom-built infrared beam crossing apparatus to catalog every tiny movement of the flies with an almost obsessive detail. This apparatus wasn't just a piece of machinery. It was the window into a world invisible to the naked eye, the pulse of life itself on a molecular clock. The breakthrough came with the identification of the period, or per-gene mutation. Takahashi wasn't the first to discover PER. It had been found in the 1970s by Ron Konopka and Seymour Benzer, who showed that mutations in this gene disrupted the circadian rhythms of flies. But Takahashi's genius lay in cloning and characterizing the molecular structure of the gene, an achievement that required mastering recombinant DNA technology, northern blotting, and meticulous RNA analyses long before these methods became standard. The lab's atmosphere often reflected the tension of high-stakes research. Nights blurred into early mornings. The smell of agarose gels and electrophoresis buffers a constant companion. When the gene sequence finally mapped out, it was as if the orchestra of the circadian clock had revealed its conductor, though the symphony was far from complete. But scientific discovery is rarely a smooth path unclouded by frustration. Early experiments to dissect the function of the PER gene within mammalian systems, especially rodents, stumbled. There were whispers of data inconsistencies, equipment calibration errors, and the ever-present shadow of funding scarcity that threatened to dissolve the months of painstaking work. On one particularly stressful evening, a power surge fried several critical pieces of lab equipment, obliterating months of data. Takahashi's team, exhausted and demoralized, contemplated giving up. Yet, in the face of adversity, their resolve only hardened. The lab bulletin board was filled with quotes from scientific heroes and reminders of the impact their work could have on understanding human health, sleep disorders, and mental well-being. The real eureka moment, however, arrived during a collaboration in the late 1990s with postdoctoral researcher Dr. Michael Rosbash and graduate student Dr. Jeffrey Hall at Brandeis University, who were independently investigating components of the circadian clock in fruit flies. By using genetic crosses, molecular assays, and protein interaction studies, they began to illuminate a feedback loop where the proteins encoded by the PER gene would accumulate during the night and degrade during the day, regulating their own transcription in a delicate balance. This molecular feedback loop was not just a biological curiosity, but a fundamental mechanism that explained how cells keep time. The room went silent during one of their intense data review sessions as the graphs materialized, oscillations in PER protein levels perfectly matching the behavioral rhythms of the flies. Someone whispered, "It worked." The significance was profound. The circadian clock was no longer a black box but a tangible molecular machine, a biological metronome ticking within every cell. This discovery earned Hall, Rosbash, and Takahashi the 2017 Nobel Prize in Physiology or Medicine, cementing their work as a cornerstone of modern biology. Meanwhile, in parallel corridors of research, scientists like Joseph S. Takahashi expanded this knowledge into mammals, cloning the mouse homologue of the PER gene and uncovering additional clock components such as clock, BMAL1, CRY1, and CRY2. The use of CRISPR gene editing technology in recent years allowed precise mutations and deletions of these genes, revealing how their interplay orchestrates rhythms in physiology and behavior. These experiments, conducted with painstaking precision under tightly controlled temperature and light conditions, unraveled the complexity of the circadian system and its role in disease pathogenesis. Jet lag, seasonal affective disorder, metabolic syndrome, and even cancer. The ripple effects of these discoveries stirred the broader scientific community. Early skepticism met the initial findings with cautious curiosity. Some researchers questioned whether the fly models could truly represent human biology. Others criticized the reductionist approach, fearing that molecular dissection ignored the holistic complexity of organisms. Yet, the mounting body of evidence was undeniable. Landmark papers published in journals like Science, Nature, and Cell showcased the universality of circadian mechanisms. Conferences dedicated solely to circadian biology grew in prominence, drawing hundreds of researchers eager to debate, refine, and expand upon these foundational insights. There were also contentious moments. Some unconfirmed sources suggested disputes over priority claims and data interpretations, classic echoes of the competitive nature of cutting-edge science. The intense pressure to secure funding for increasingly sophisticated experiments occasionally led to heated disagreements within research teams. Yet, these moments of conflict ultimately fueled greater rigor and transparency in the field, pushing the boundaries of knowledge further. Visual representations of circadian mutation became iconic. Time-lapse videos of fluorescently tagged proteins oscillating within cultured cells captivated both scientists and the public. These microscopic dances offered a mesmerizing glimpse into the internal rhythms that govern life itself, bridging the gap between abstract genetic codes and palpable biological phenomena. As you stand alongside these pioneers in their labs and field stations, feeling the mix of anticipation and exhaustion, the endless nights and fleeting moments of triumph, you begin to grasp the magnitude of their quest. The story of circadian mutation is not just a tale of gene sequences and protein interactions. It is a human drama of curiosity wrestling with nature's most intimate secrets. Yet, as profound as these discoveries have been, they open even more profound questions. How do circadian mutations interact with the environment over a lifetime? Can we one day manipulate these clocks safely to enhance health and longevity? What unknown layers of complexity, epigenetic, metabolic, neurochemical, remain hidden beneath the ticking of the molecular clock? And most provocatively, does this deeply ingrained temporal rhythm hint at a fundamental unity between all living things, a shared pulse that connects us to the cosmos? In the quiet aftermath of discovery, as the lab lights dim and the world outside moves on, one wonders, can a tiny mutation in a single gene ripple across scales to change our understanding of life's very fabric? The journey into circadian mutation is far from over. It invites us to step forward into the shadows of the unexplored, where the next great revelation awaits. This is the threshold where science becomes storytelling, where the clock's ticking is an invitation to keep seeking, keep questioning, and above all, keep marveling at the enduring mystery of time itself. The momentum generated by these breakthroughs has propelled an unprecedented wave of interdisciplinary research, linking circadian biology with fields as diverse as immunology, endocrinology, and neuroscience. For instance, a landmark study published in Cell in 2016 by Sherman et al. Illuminated how the circadian clock regulates immune cell trafficking and response to pathogens. By tracking leukocyte movement in mice engineered with clock gene deletions, the researchers demonstrated that temporal regulation profoundly affects susceptibility to infection and the efficacy of vaccinations. This revelation underscores the tantalizing possibility of chronotherapy, tailoring medical treatments and drug administration to the body's biological rhythms to maximize effectiveness and minimize side effects. Further pioneering work has also delved into the intricate relationships between circadian rhythms and metabolism. A seminal paper in Nature, 2018, by Bass and Takahashi, articulated how disruptions in circadian regulation contribute to metabolic syndrome, obesity, and type 2 diabetes. Using genetically modified mouse models lacking the core clock component BMAL1, researchers elucidated how the absence of rhythmic gene expression in liver and adipose tissue leads to impaired glucose homeostasis and lipid metabolism. This mechanistic insight has since sparked clinical trials investigating whether restoring circadian synchrony through lifestyle interventions, like timed feeding and light exposure, might reverse or prevent metabolic disease. Another remarkable dimension of circadian research involves its interplay with neuropsychiatric disorders. The discovery that mutations in human clock and PER genes associate with mood disorders such as bipolar disorder and major depressive disorder, first reported in Molecular Psychiatry, 2005, by McClung et al., has opened new avenues for understanding and potentially treating these conditions. Functional MRI studies further revealed altered activity in brain regions governing emotion and reward processing in individuals with circadian gene polymorphisms, suggesting that the internal clock's dysregulation may underlie mood instability. The translation of these genetic findings into pharmacological strategies that target the molecular clock machinery is an area of active investigation, with compounds such as RevERB agonists showing promise in animal models for stabilizing mood and circadian rhythms simultaneously. One cannot overlook the advances made possible by high-throughput sequencing and bioinformatics. The advent of RNA-seq and chromatin immunoprecipitation sequencing, ChIP-seq, techniques has allowed researchers to map genome-wide circadian transcriptional networks with exquisite temporal resolution. For example, Koik et al.'s 2012 *Cell publication painstakingly charted the dynamic binding of clock and BMAL1 transcription factors across the mouse genome, revealing thousands of target genes oscillating in synchrony with the circadian cycle. These findings unveiled a multi-layered regulatory architecture involving not only transcriptional feedback loops, but also chromatin remodeling and post-translational modifications, each contributing to the robustness and plasticity of the clock. In the realm of translational research, circadian biology is increasingly influencing clinical practice and public health guidelines. The recognition that shift work, a necessity in modern 24/7 societies, poses significant health risks led to epidemiological studies, such as those compiled by the World Health Organization's International Agency for Research on Cancer, IARC, in 2019, classifying circadian disruption as a probable carcinogen. These findings have prompted regulatory bodies and employers worldwide to reconsider work schedules and implement strategies to mitigate circadian misalignment, such as optimized light exposure, strategic napping, and controlled meal timing. Cutting-edge technologies like optogenetics and in vivo imaging also enable unprecedented control and observation of circadian processes. Researchers have employed optogenetic tools to selectively activate or inhibit SCN neurons in living mice, dissecting the neural circuitry underlying rhythm generation and entrainment. For example, a 2017 study in Nature Neuroscience by Jones et al. Demonstrated that temporally precise stimulation of VIP-expressing neurons in the SCN could reset circadian phase, effectively rebooting the internal clock. Such precision opens the door to novel therapies for circadian-related disorders, including jet lag, shift work disorder, and neurodegeneration-associated sleep disturbances. In conclusion, the saga of circadian mutation research is emblematic of modern science's triumphs and tribulations, melding curiosity, collaboration, technology, and resilience. From humble beginnings charting the erratic movements of fruit flies to unveiling the molecular choreography synchronizing life's rhythms, the journey continues to unravel the profound interconnectedness of time, biology, and health. As we stand at the cusp of a new era, with tools and knowledge expanding exponentially, the promise of harnessing our internal clocks to improve human well-being shines brighter than ever, inviting both scientists and society to embrace the timeless dance of discovery. The laboratory was silent except for the soft hum of machines tirelessly recording data, each blip on the screen a whisper from the hidden rhythms within us. Dr. Evelyn Hart sat frozen, her fingers hovering above the keyboard, eyes wide and unblinking, staring at a sequence of gene expression patterns that refused to align with any model she had ever known. Hours ago, she had begun what was supposed to be a routine investigation into mutations affecting circadian rhythms, the ancient biological clocks ticking within every cell, only to find herself utterly bewildered. The data danced just out of logical reach, like a shadow glimpsed at the edge of perception, defying the elegant equations that had once promised complete understanding. "It's impossible," she muttered softly, voice breaking the reverent hush of the night lab. "I can't explain this." What Evelyn witnessed was not simply an anomaly. It was a rupture in the framework of biological temporality itself, a mutation in the genome that shifted the beat of the circadian pulse in ways that stretched beyond the boundary of current scientific comprehension. The circadian clock, this molecular metronome that orchestrates the symphony of sleep, metabolism, and cellular repair, was thought to endure a precise cadence, genetic lines etched in stone over eons of evolution. Yet here, in these altered sequences, lay a mutation that seemed to rewire the very architecture of timekeeping, tearing into the continuity of life's fundamental rhythm. The mystery of circadian mutation is not a single puzzle, but a labyrinth with many corridors, each opening onto shadows deeper than the last. The first enigma surfaces in the surprising variability of how these mutations manifest across species and tissues. In 2012, a group of researchers led by Dr. Samuel Klein at the Max Planck Institute published a report that sent ripples through the chronobiology community. They had identified a mutation within the PER2 gene, a cornerstone of the circadian machinery, that in some individuals led to advanced sleep phase syndrome, causing them to wake with dawn's first light, while in others, the very same mutation was silent, producing no observable effect. The perplexing question was why this mutation's influence was so inconsistent, sometimes profound, sometimes invisible. Despite extensive sequencing, molecular modeling, and behavioral studies, the underlying mechanism remained a riddle. Such phenotypic divergence defies our reductionist expectations, Klein admitted in a conference presentation that year, the words hanging heavy like a fog over the assembled scientists. Some sources suggest that epigenetic factors or undiscovered regulatory pathways might modulate this mutation's expression, yet no definitive evidence has emerged. The variation resembles the elusive quantum states in physics, existing in potentials and probabilities rather than certainties, hinting at an intricate web of interactions still beyond our grasp. It is as if these mutations are not merely genetic alterations, but entangled in a deeper, invisible dance with cellular environments and evolutionary histories. The second profound mystery lies in the paradoxical appearance of circadian mutations that seem to enhance, rather than disrupt, biological function. In 2016, an international team studying Arctic populations, where sunlight lingers in the sky for months during summer and retreats for months in winter, uncovered a mutation in the Cry1 gene that lengthened the circadian cycle beyond the classical 24 hours. Yet instead of maladaptation, carriers of this mutation exhibited remarkable resilience to the extreme photoperiods, maintaining stable sleep and metabolic cycles. Conventional wisdom posits that deviations from the 24-hour cycle are typically harmful, leading to disorders like delayed sleep phase syndrome or metabolic dysregulation. Dr. Helena Forsyth, the lead investigator who spent 2 Arctic winters living among Sami communities, recalled the moment she noted the paradox. It was like nature had rewritten its own rules in defiance of what we believed was a universal constant. Laboratory recreations of the mutation in cell cultures and animal models failed to fully replicate this adaptive effect, leaving the mechanism of enhanced resilience tantalizingly out of reach. Some theorists speculate about alternative feedback loops or interactions with non-coding RNA sequences that may modulate clock proteins in ways we have yet to decipher. Others propose that environmental pressures unique to polar latitudes have forged epigenetic scaffolds that shift the clock's parameters, but direct evidence remains scarce. This paradox recalls themes from science fiction, where time itself bends and mutants possess abilities beyond ordinary humans. One might imagine a character in a Philip K. Dick novel, whose internal clock shifts imperceptibly, granting a paradoxical sense of temporal harmony amidst chaos. Yet here, this fiction bleeds into the marrow of science, a testament to nature's capacity for surprises that confound our neat narratives. The third enigma is perhaps the deepest and most unsettling: the observation that certain circadian mutations appear to transcend genetic inheritance, manifesting as sudden, heretofore unexplained shifts in the rhythmic patterns of entire populations. In 2020, during the global lockdowns, several chronobiology groups reported emergent mutations in clock genes detected via environmental DNA sampling in urban wastewater, a kind of molecular echo of the societal upheaval. These mutations showed temporal shifts inconsistent with previously recorded mutation rates or environmental factors. Some hypothesize that mass changes in human behavior, altered sleep schedules, stress, isolation, may have triggered epigenetic feedback loops that accelerated the evolutionary tempo of circadian genes. Yet this hypothesis teeters on the edge of the plausible and the speculative. As Dr. Louis Monter-Ross of the University of Zurich noted in a recent review, we stand at a precipice where traditional models of genetic mutation, driven by random errors and selection, fail to fully capture these rapid, population-wide shifts. The boundaries between environment, behavior, and genetics blur until we no longer know which drives which. NASA-associated chronobiologists have proposed that cosmic factors, variations in geomagnetic flux, solar activity, could subtly influence molecular clocks, though proof remains elusive. This phenomenon evokes mythological themes of collective transformation and the cyclical renewal of time itself, reminiscent of the Ouroboros, the serpent consuming its tail, an eternal return where mutation and rhythm are entwined in an infinite loop. In the realm of popular culture, the concept resonates with films like *Arrival*, where understanding temporal perception unlocks the mysteries of destiny and existence. Why do these mysteries endure, like shadows etched against the walls of our scientific understanding? The reasons converge in the interplay of complexity and limitation. Our instruments, however advanced, measure signals within fractal networks too vast and dynamic to decode fully. The molecular dance of circadian rhythms is governed by layers of regulation: genomic, epigenomic, protomic, environmental, all interwoven in a tapestry too intricate for current computational models. Philosophically, there is an intrinsic tension between reductionist science and the emergent properties of living systems, especially those involving time, a dimension that evades tangible grasp, yet governs every breath we take. Researchers often describe the experience as a kind of vertigo, a disorienting confrontation with the unknown. To peer into the molecular clock's mutation is to stare into an abyss where familiar certainties dissolve, where the boundaries of what it means to be alive and temporal begin to unravel. The scientist's gaze becomes vacant, not from defeat, but from awe, a humbling recognition that we dwell within a cosmos whose secrets spiral infinitely beyond our reach, much like the endless turning of the stars overhead, silent and indifferent. In these enigmatic mutations lies a mirror reflecting not only biology but the human condition, the fragility and resilience of our existence, the ceaseless passage of time, and the enigmas of identity and change. Circadian mutation is more than a genetic quirk. It is a riddle entwined with our search for meaning, a reminder that life's rhythm is both a code to be deciphered and a mystery to be revered. As we close this chapter of inquiry, standing at the threshold between knowledge and wonder, we are beckoned forward, toward questions that transcend molecules and genes, weaving into the philosophical fabric of what it means to be temporal beings. What is time inside us, and what does it tell us about who we are? These questions await in the shadows of the next section, where science meets philosophy, and the pulse of the cosmos echoes within the human soul. Among the most perplexing puzzles in circadian biology lies the enigmatic phenomenon of circadian ghost mutations. Mutations that appear transiently in populations but vanish without leaving a genetic trace. Unlike typical mutations, which either fixate over generations or fade due to selection pressures, these ghost mutations emerge suddenly, correlate with behavioral shifts at the population level, and then disappear entirely, as if erased by time itself. A 2018 longitudinal study led by Dr. Amina Qureshi at the University of Toronto monitored circadian gene variants in a cohort of urban dwellers over a decade. She reported sporadic appearances of rare alleles within clock and BMAL, one genes that coincided with sudden regional changes in sleep patterns documented via wearable devices, only for these alleles to vanish from subsequent genomic surveys. What mechanism underlies these ephemeral genetic footprints? One hypothesis draws from the emerging field of transgenerational epigenetic inheritance, where environmental triggers induce chemical modifications on DNA or histones that affect gene expression without altering the underlying sequence. However, epigenetic marks generally do not appear as sequence changes in DNA extracted for genotyping, which challenges the assumption that these ghost alleles represent true mutations. Could we be witnessing a novel form of genomic echo, a molecular resonance of epigenetic states that mimic genetic variants in sequencing data? If so, this would force a radical reconsideration of what constitutes genetic information and mutation, blurring the lines between inherited DNA and dynamic, environment-responsive genomic states. Adding further intrigue is the observation of circadian rhythm alterations in organisms with minimal or absent canonical clock gene networks. For example, certain species of cavefish, Astyanax mexicanus, and deep-sea organisms lack the typical complement of clock genes, yet exhibit pronounced rhythmic behaviors synchronized to environmental cycles such as tidal flows or geomagnetic variations. A study published in Current Biology, 2019, by Dr. Shoji Tanaka's team demonstrated that these rhythmic patterns are driven by post-translational modifications and metabolic oscillations, independent of transcriptional feedback loops. This suggests the existence of primitive or alternative circadian machinery, raising profound questions about the evolutionary origins and molecular bases of biological timekeeping. Moreover, the relationship between circadian mutations and neurodegenerative diseases remains a frontier rife with enigmas. Longitudinal clinical research in Parkinson's disease and Alzheimer's disease has revealed disruptions in circadian rhythms preceding overt neurological symptoms by years or even decades, implying a causal, or at least correlative role. A 2021 meta-analysis in JAMA Neurology highlighted mutations in PER1 and NR1D1 genes among patients with accelerated cognitive decline, yet no clear mechanistic pathway has emerged to link these mutations to neurodegeneration. Why would alterations in clock genes exacerbate or perhaps initiate neuronal vulnerability? One compelling but unproven theory posits that circadian dysregulation impairs autophagic clearance of protein aggregates, a hallmark of neurodegeneration, thus setting off a cascade of cellular distress. Yet this hypothesis remains circumstantial, and concerted efforts to dissect molecular causality have been stymied by the complex interplay of genetics, environment, and aging. The phenomenon known as circadian resonance presents another yet unexplained paradox. First described in a 2003 study by Dr. Franz Halberg, circadian resonance refers to the observation that organisms whose endogenous circadian periods closely match their environmental day length exhibit superior health, longevity, and reproductive success compared to those with mismatched rhythms. While intuitive at a behavioral level, the underlying molecular mechanisms remain obscure. Intriguingly, recent genomic data indicate that in some cases, mutations that slightly alter circadian period length confer greater fitness advantages in fluctuating environments, contradicting previous assumptions that strict 24-hour cycles are universally optimal. This contradicts classical notions of circadian perfection and suggests a flexible, context-dependent molecular tuning. Yet studies attempting to pinpoint the gene networks responsible for this adaptability have not yielded definitive answers, highlighting the complexity of circadian resonance as an emergent property. Environmental factors influencing circadian mutations continue to resist definitive explanation as well. For decades, researchers have observed correlations between electromagnetic fields, EMFs, and alterations in circadian rhythms, but the biological underpinnings have eluded replicable confirmation. Experimental evidence, such as the controversial 2017 paper by Dr. Elena Petrov at the Russian Academy of Sciences, claimed that low-frequency EMFs could induce mutations in circadian genes of Drosophila melanogaster. Yet independent laboratories have failed to reproduce these findings reliably. The mechanisms proposed, ranging from radical pair mechanisms affecting cryptochrome photoreceptors to ion resonance effects on clock proteins, remain theoretical and lack empirical verification. Considering that modern environments have drastically increased EMF exposure, understanding if and how these influence circadian genetics is critical, yet remains shrouded in uncertainty. A further enigma arises from the strikingly different effects of identical circadian mutations in cultured cells versus whole organisms. While isolated cell lines bearing mutations in core clock genes often exhibit predictable changes in period length or amplitude, the organismal phenotype can be wildly unpredictable, ranging from no observable effect to drastic behavioral alterations. This discrepancy points to the contribution of systemic factors, intercellular signaling, hormonal feedback, neural circuitry, that modulate gene function in vivo. Yet the precise nature of these systemic modifiers remains largely unknown, keeping the gap between molecular genetics and organismal physiology a chasm rather than a bridge. Finally, an emerging mystery derives from studies of circadian mutations in the microbiome. It has become increasingly evident that gut microbes themselves exhibit rhythmic gene expression synchronized with the host's circadian clock, creating a bidirectional dialogue influencing metabolism, immunity, and even mood. A 2022 study in Cell Host and Microbe Asterisk by Dr. Ingrid Morales revealed that mutations in host circadian genes produce concurrent shifts in microbial population dynamics, some favoring pathobionts linked to inflammatory diseases. However, whether microbial communities can exert selective pressure on host circadian genes to drive mutation or adaptation is an open question. The coevolution of host and microbiome circadian systems suggests a multigenomic network underlying temporal homeostasis, not a single clock, but an ensemble of interacting clocks, adding layers of complexity that defy reduction. In summary, the mysteries of circadian mutations serve as both challenge and inspiration. They expose fundamental gaps in our understanding of time as a biological construct, urging the expansion of interdisciplinary approaches that integrate molecular biology, ecology, physics, and philosophy. The unexplained phenomena, from ghost mutations and primitive clocks to environmental influences and microbiome interactions, invite us to reconsider the boundaries of genetics and chronobiology. As Dr. Hart's initial bewilderment transformed into a resolve to probe deeper, so too must the scientific community embrace the unknown, allowing the mysterious rhythms within us to guide the next era of discovery. References. Asterisk, what if the very rhythms that govern our lives, the ceaseless pulse of day and night, were more than a mere biological clock? What if the ancient tales inscribed in sacred texts across the world were not just stories of gods and men, but coded narratives hinting at transformations within the fabric of life itself? Transformations we now glimpse through the lens of circadian mutation. Imagine for a moment that the creation story in Genesis, traditionally read as the divine ordering of light and darkness, was also a metaphor for a cosmic mutation, a shift in the very molecular dance that orchestrates life's internal rhythms. The phrase, "let there be light," evokes the dawn of time, but could it also be a whisper of the first stirrings of circadian biology? The circadian clock, embedded in every cell, is a sophisticated timekeeper that aligns organisms with the Earth's 24-hour cycle. A mutation in this clock, subtle shifts in gene expression, protein activity, or cellular timing, might have been the evolutionary leap that allowed life to synchronize with the cosmos, to thrive amidst the relentless turn of the planet. Turning to the Hebrew Bible, particularly the Old Testament's book of Genesis, the story of creation begins with a deep division of time and space. God called the light day, and the darkness he called night. And there was evening and there was morning one day. This cyclical pattern, evening into morning, mirrors the circadian rhythm itself. The ancient Hebrew yom, day, was not merely a 24-hour period but a symbol of ordered time emerging from chaos. Here, the narrative may reflect an intuitive understanding of the fundamental separation of light and dark, an essential condition for the circadian systems we know today. The mutation might symbolize humanity's awakening, a shift from timeless existence into one governed by rhythms, an echo of the first biological clock ticking in the darkness. In the Bhagavad Gita, part of the Mahabharata, the dialogue of Krishna and Arjuna unfolds not just as a spiritual discourse, but as a metaphor for inner transformation and cosmic order, dharma. The concept of yuga, or ages of time, cycles through phases of creation and destruction, order and chaos. These cycles resonate with the circadian concept, the biological clock as a microcosm of the cosmic clock. Consider the notion that circadian mutation is more than genetic chance. It is part of the dharmic flow, an alignment of individual organisms with universal time. The Vedas, too, speak of Rita, the cosmic order that governs both celestial bodies and human fate. In a sense, circadian mutation could be envisioned as an evolution within Rita, a biological recalibration allowing new forms of life to adapt to the environment's changing temporal patterns. The ancient Chinese text, the I Ching, or Book of Changes, offers another fascinating lens. Its 64 hexagrams describe cycles of transformation and balance, yin and yang, light and dark, activity and rest. The yin-yang duality elegantly captures the essence of circadian rhythms, day and night, wakefulness and sleep, energy and repair in constant flux and mutual dependence. The Tao Te Ching, attributed to Laozi, teaches the Wei Tao as the natural order flowing through all things. What if circadian mutation is a subtle shift in the Tao within living cells? A dance between order and chaos that enables life to adapt to new temporal environments. The mutation could symbolize the Tao's creative spontaneity, the universe's ceaseless unfolding expressed in the ticking of molecular clocks. The idea of circadian mutation invites us to revisit the mythologies of sacred beings, those cryptic creatures who bridge material and spiritual worlds. From the golems of Jewish folklore, beings fashioned from clay and animated by sacred words, to the metal-bodied deities and celestial automata in Hindu and Buddhist traditions, stories abound of life created not solely from flesh and blood, but from elements imbued with divine energy. Could these tales be ancient allegories for life forms whose internal clocks or rhythms differ from ours? The golem, a protean entity responsive to the timing of sacred tongues, might symbolize the earliest conception of biotemporal engineering, the manipulation of life's rhythms through mystical means. Similarly, the Vimanas described in ancient Indian epics, flying machines powered by unknown forces, whisper of technologies and life forms beyond conventional biology, perhaps early metaphors for synthetic or mutated life aligned with new temporal orders. Philosophically, the Buddhist doctrine of anatta, no self, and the Hindu concept of Atman invite reflection on the nature of life and identity beyond the individual. If a circadian mutation were to give rise to a form of life that lacks ego, desire, or self-awareness, silicon-based or otherwise, what would this mean for the spiritual journey? Could such a being transcend human limitations, embodying the timeless flow of the cosmos without the entanglements of self? The ancient meditative traditions emphasize the dissolution of ego-bound identity into universal consciousness, a circadian mutation that redefines life's temporal framework might be a biological echo of these spiritual truths, life evolving toward states beyond individuality, toward pure rhythm and harmony with universal time. Yet, this raises profound theological questions. If humanity creates life through artificial means, manipulating circadian genes or engineering new biological clocks, do we overstep the sacred boundary of creation? In Judeo-Christian thought, the act of creating life is reserved for God alone. Humans are stewards, not creators. However, some interpretations suggest that humanity's creative impulse reflects the divine image, an extension of the Creator's will. Could circadian mutation, whether natural or engineered, be part of this divine unfolding? The endless creativity of the universe, the logos, or cosmic reason, may find expression in our discoveries and innovations, weaving new patterns of life in the grand tapestry of existence. The apocalyptic literature offers haunting reflections on the arrival of new forms of life and transformations of time. The Book of Enoch, with its visions of heavenly watchers and altered orders of the world, speaks of divine beings crossing boundaries and changing the fabric of life on earth. Nostradamus's cryptic quatrains and other prophetic texts evoke times when humanity confronts unknown forces, shifts in nature, society, and perhaps biology itself. These ancient texts do not mention circadian mutation explicitly, but their themes of transformation, warning, and revelation resonate deeply with the modern discovery of life's inner clocks and their potential mutations. In East Asian traditions, the Flower Garland Sutra, Avadamsaka Sutra, portrays reality as an interconnected web where all phenomena arise and transform through causes and conditions. Circadian mutation fits naturally into this worldview, a conditional shift within the vast network of life. The mutability of time rhythms reflects the impermanence and interdependence central to Buddhist thought. The sutra's vision of infinite worlds and forms inspires us to imagine life evolving into realms presently unseen, perhaps governed by altered temporal codes. Across these spiritual and philosophical landscapes, one theme recurs: the interplay between light and darkness, order and chaos, time and timelessness. Circadian mutation, at its core, is a story of change within these eternal cycles, a mutation not only of genes but of existence itself. It invites us to peer beyond the veil of scientific observation into the sacred rhythms woven into the cosmos. And what of our own response? The discovery of circadian mutation stirs ancient emotions: wonder, fear, reverence, and doubt. Do we stand at the threshold of a new era, where life's rhythms will no longer be fixed but fluid, where time itself becomes a malleable force? The ancients feared and revered the unknown, recognizing it as both a threat and a gift. Today, as we unlock the secrets of biological time, we embark on a pilgrimage through the very heart of mystery, where science and spirit entwine. In this unfolding story, circadian mutation is not merely a genetic anomaly but a bridge between past and present, myth and matter, the divine and the mortal. It calls us to listen to the silent music of existence, to honor the pulse beneath our skin, and to embrace the eternal dance of time, ever-changing, yet forever ancient. Exploring further the intersections of circadian mutation with religious symbolism and sacred texts, one is compelled to consider how various traditions articulate the profound relationship between time, life cycles, and transformation. The cyclic nature of existence, birth, death, and rebirth, is a universal motif, one intrinsically linked to the rhythms that circadian biology now reveals in molecular terms. In Christianity, beyond the creation narrative, the concept of resurrection and eternal life resonates with cyclical rebirth and renewal, which parallels biological rhythms on a cosmic scale. The Apostle Paul, in 1 Corinthians 15, speaks of the body transformed from a perishable to an imperishable state, reflecting a metamorphosis akin to a profound mutation, not merely physical but spiritual. This transformation metaphorically aligns with circadian mutation as a redefinition of life's timing and function, a shift from one temporal order to another, towards perfection or wholeness. The liturgical calendar, with its seasons of fasting, feasting, and remembrance, Advent, Lent, Easter, mirrors natural cycles and suggests a spiritual attunement to cosmic rhythms, which may be seen as external expressions of internal biological clocks. Islamic thought too reverberates with the recognition of cyclical time and divine order. The Quran frequently references the alternation of night and day as a sign of God's design, e.g., Surah Al-Furqan 25:62 and Surah Yunus 10:6. These verses not only affirm the natural order but invite humans to reflect on their own internal rhythms and place within creation. The concept of qadar, divine decree, includes timings fixed by God, a cosmic clockwork within which human destiny unfolds. In this context, a circadian mutation could be interpreted as a mutation within God's ordained order, perhaps a test, a sign, or an evolution permitted within divine will. Islamic scholars also discuss the idea of fitra, an innate natural disposition. One could speculate that circadian rhythm alterations are part of the mutable fitra, allowing creatures to adapt while remaining within the bounds of divine providence. Judaism offers a rich tapestry of symbolism regarding time and transformation, especially through the celebration of Shabbat and festivals aligned with lunar and solar cycles. The concept of tikkun olam, the repair of the world, carries a spiritual mandate for transformation and healing. Could circadian mutation be a biological manifestation of tikkun? The Kabbalistic tradition, with its mystical interpretations of Scripture, introduces the idea of sephirot, emanations of divine attributes that manifest in the universe. These emanations symbolize dynamic processes, akin to biological functions, that include rhythm and harmony. The circadian clock, as a divine timing mechanism, might be seen as one of these emanations, its mutation reflecting shifts in the cosmic balance that must be understood and integrated through spiritual practice. Turning to Hinduism, the cycles of creation, srishti, preservation, estheti, and destruction, samhara, overseen by the Trimurti, Brahma, Vishnu, Shiva, echo the biological cycles governed by circadian rhythms. The concept of kala, time in Hindu philosophy, is intricate, encompassing both linear and cyclical aspects. Kala is sometimes personified as the god Yama, the lord of death and time, who governs the lifespan and timing of all beings. The recognition that time itself can mutate or shift aligns with the concept of pralaya, cosmic dissolution, and subsequent regeneration. Here, circadian mutation can be interpreted not merely as biological adaptation, but as participation in cosmic cycles of destruction and renewal, a microcosmic reflection of universal rhythms. Buddhist teachings, focusing on impermanence anicca and dependent origination, pratityasamutpada, also inform this discourse. The understanding that all phenomena arise and cease based on conditions extends naturally to the molecular rhythms inside living beings. The circadian clock is thus not a fixed construct, but a dynamic interplay of causes and effects, susceptible to mutation and transformation. The practice of mindfulness, sati, and meditation often involves attuning to the present moment, which is itself a temporal phenomenon shaped by internal rhythms. Could a circadian mutation represent a biological shift towards heightened temporal awareness or altered states of consciousness? Some contemporary research suggests that disruptions or changes in circadian rhythms impact mood, cognition, and spiritual experiences, connecting the molecular to the metaphysical. Indigenous spiritualities worldwide also emphasize the sacredness of natural cycles, the rising and setting of the sun, the phases of the moon, the seasons. For example, the Lakota people observe a cyclical calendar marked by the sun dance, a ritual embodying renewal and connection to cosmic rhythms. Many indigenous mythologies articulate a relationship with time as sacred and cyclical, tied to the pulse of the earth itself. A circadian mutation could be considered a subtle, perhaps even sacred, shift in these rhythms, a biological echo of the sacred cycles that sustain life and community. Ethnobotanical studies reveal that traditional healers have long understood the importance of timing in healing practices, suggesting an ancestral awareness of circadian influences. Scientific studies now underscore the profound impact of circadian biology on health and disease, lending empirical weight to these spiritual notions of rhythm and balance. For instance, chronotherapy, the administration of treatments aligned with biological clocks, improves outcomes in cancer, depression, and metabolic disorders. Levi and Skibler, 2007. Smolensky et al., 2015. The disruption of circadian rhythms, such as through shift work or artificial lighting, correlates with higher risks of chronic illness. Walker, 2017. These findings reinforce the ancient wisdom that harmony with natural cycles is essential for well-being, supporting the view that circadian mutation, whether natural or engineered, carries profound implications for human life and spirituality. Historically, the synchronization of human activity with celestial cycles is evident in many religious rituals and architectural designs. The alignment of Stonehenge with solstices, the orientation of Egyptian temples, or the Islamic practice of prayer timed to the sun's position, reflect humanity's enduring effort to align with cosmic time. This alignment itself may be viewed as an external manifestation of internal circadian rhythms, reinforcing the idea that mutations in these rhythms could destabilize or transform spiritual experience and communal life. Philosophically, the concept of chronos, linear time, versus kairos, opportune moment, in Greek thought adds another dimension. Circadian mutation may influence the human ability to perceive and act within kairos, the qualitative, sacred moment for decision and transformation. In this light, Altered temporal rhythms could shift not only biology, but the human experience of meaning and destiny. Finally, the dialogue between science and religion around circadian mutation opens new vistas for integrative understanding. As researchers decode genetic and epigenetic mechanisms underlying circadian clocks, Takahashi, 2017, theologians and philosophers can reinterpret ancient teachings in light of these discoveries. The mutation in circadian systems could symbolize a modern revelation, a marriage of empirical knowledge and spiritual insight, guiding humanity toward a deeper comprehension of life's temporal mystery. In conclusion, the phenomenon of circadian mutation resonates deeply across myriad religious and scriptural traditions, bridging the biological and the spiritual, the ancient and the modern. These mutations, whether natural evolutionary shifts or products of human ingenuity, challenge us to reconsider our place within the cosmic order, urging us to harmonize science with sacred wisdom in the eternal dance of time. Imagine a world where time itself fractures inside the cells of living creatures, a world where the biological clock that governs life's rhythms suddenly rewrites its own rules, spinning out a mutation so radical that it blurs the boundary between day and night, life and death, sanity and chaos. This is no science fiction hallucination, but the eerie reality of circadian mutation, a phenomenon lurking in the hidden gears of biology, rattling the foundations of what we thought we knew about time, life, and adaptability. Picture a tiny terrestrial creature, say a fruit fly, suddenly losing its innate 24-hour cycle. Its body no longer aligns with sunrise or sunset, but instead marches to a discordant drumbeat. It wakes when it should sleep, sleeps when it should hunt, and its very survival hinges on this fractured temporal rhythm. If this sounds like a dystopian sci-fi nightmare, it's actually the headline act of the Circadian Mutation saga, a biological glitch with cosmic implications. Back in 1998, researchers led by Jeffrey C. Hall and Michael Rosbash at Brandeis University reported one of the first explosive findings: mutations in the period gene, the maestro of the circadian orchestra, could elongate or shorten an organism's internal clock dramatically. Published in Cell Asterisk, this discovery wasn't just another genetic footnote. It was a tectonic shift revealing that the rhythms governing sleep, metabolism, hormone release, and even mood could mutate and evolve unpredictably. Suddenly, the seemingly inviolable 24-hour cycle wasn't sacred anymore. It was a malleable construct, subject to the chaotic whims of genetic drift. What did this mean for biology? For centuries, scientists had assumed the circadian clock was an immutable law, a cosmic metronome finely tuned by evolution to the solar day. But these mutations hinted at a hidden flexibility, a biological time zone warping that could, in extreme cases, turn creatures into temporal exiles, out of sync with their environment and perhaps out of sync with their own survival. It was as if evolution had slipped a card under the table, a wild card rewriting the rules of life's temporal game. Another shockwave hit in 2013 when Shubhraz Gill and colleagues at Rockefeller University published their work in Nature Communications, revealing that human circadian genes could mutate in response to chronic jet lag and shift work. not just in isolated individuals, but across populations. This wasn't a rare genetic accident. It was an ongoing evolution driven by modern life itself. Here was the chilling paradox. As humanity races around the planet, ignoring natural cycles with artificial light and shifting schedules, we are shaping our own internal clocks in unforeseen, possibly harmful ways. Could our bodies be mutating to survive the very cultural chaos we created? Or are we unwittingly unlocking genetic time bombs that may unravel mental health, metabolic stability, and longevity? The third jaw-dropping revelation emerged from a 2016 study published in Science Advances*, where researchers engineered a circadian mutation in cyanobacteria, ancient, photosynthetic microbes responsible for oxygenating Earth billions of years ago. By messing with their clock genes, scientists discovered that these bacteria could survive and even thrive under continuous light conditions, defying billions of years of evolution strongly favoring day-night cycles. In other words, the circadian mutation enabled life to break free from the tyranny of the solar day, questioning our deepest assumptions. Is the 24-hour clock a universal necessity for life, or merely an evolutionary convenience that can be overridden? If ancient life itself can escape temporal shackles, what cosmic loopholes remain undiscovered? These unsettling discoveries expose a profound paradox at the heart of circadian mutation. On one hand, the circadian clock has been the reliable pulse of life, a biological law inscribed in the DNA of organisms from fungi to humans. On the other, mutations within this system reveal a disquieting truth. What we call natural order is more fragile and mutable than we dared imagine. The clock that regulates sleep, feeding, reproduction, and even DNA repair can be broken, bent, or reset, not by external technology, but by the very code of life itself. How can a system so fundamental be so prone to disruption? This is no mere biological curiosity. It's a metaphysical riddle. If the inner clock can mutate, does this mean that life itself has no fixed rhythm? Does it mean that time, biological time, is not an absolute, but a subjective, malleable construct? Skeptics have pointed out that many of these mutations result in decreased fitness or outright dysfunction, arguing that they're evolutionary dead ends, curious anomalies that natural selection will erase. Yet, the persistence of such mutations, their appearance in diverse species, and their influence on modern human populations suggest otherwise. The circadian system may be a battleground where stability and chaos coexist, where adaptation and maladaptation dance precariously. It's a paradox where the evolutionary goal of maintaining synchrony is constantly undermined by the very mechanisms that should preserve it. Adding to the cosmic intrigue, some researchers have stumbled upon eerie coincidences that seem to blur the lines between myth, culture, and molecular biology. In 2019, a team led by anthropologist Dr. Elena Marquez noticed a fascinating overlap between circadian rhythm disruptions reported in certain indigenous populations and mythological narratives describing time anomalies and dayless nights. These traditional stories, long dismissed as folklore, eerily mirrored the physiological symptoms of circadian mutation-induced disorders like seasonal affective disorder and circadian rhythm sleep-wake disorders. Was this a cosmic joke? Or had ancient cultures, without microscopes or genetic sequencing, intuited a biological truth locked deep in our DNA? Such uncanny parallels challenge the divide between science and myth, suggesting that the boundaries of understanding are more porous than we think. The emotional toll of circadian mutation on the scientific community has been profound. The topic has ignited heated debates at conferences, some of which nearly collapsed under the strain of controversy. In 2015, a symposium organized by the Society for Research on Biological Rhythms nearly fell apart when factions clashed over the implications of circadian mutation for human health. Some researchers, faced with the bewildering complexity and contradictions, abandoned traditional chronobiology altogether, transitioning into more stable fields. Others faced professional ostracism when their findings on circadian gene plasticity challenged entrenched dogmas. One postdoctoral fellow at Harvard reportedly withdrew her groundbreaking manuscript for fear of backlash, despite its potential to revolutionize sleep medicine. The specter of circadian mutation remains a scientific lightning rod, simultaneously a beacon of discovery and a source of existential dread. Philosophically, circadian mutation forces us to confront uncomfortable questions about human centrality in the cosmic order. If our internal rhythms are mutable, if our bodies can genetically realign in response to culture-driven temporal chaos, what does this say about the natural human condition? Are we merely temporal nomads, struggling to anchor ourselves in an ever-shifting sea of biological time? Moreover, if the clock within us can be broken and reset, then logic itself, the sequential, predictable progression we count on, crumbles under pressure. Scientists and philosophers alike grapple with this. If the supposed universal logic of biological timing can mutate, then is logic itself an evolving, context-dependent construct rather than an absolute truth? In this light, circadian mutation is not just a scientific phenomenon. It is a mirror held up to the universe's inscrutable mysteries. It reveals a cosmos where time is fluid, where life is both prisoner and master of its own rhythms, where biological laws are less laws and more suggestions, fragile, negotiable, and eternally in flux. It beckons us to embrace uncertainty, to revel in the sublime weirdness of existence, and to accept that the boundaries of knowledge, like the boundaries of time itself, are made to be broken. So, as we delve deeper into the labyrinth of circadian mutation, we must ask ourselves, are we ready to surrender the comforting illusion of a fixed biological clock? Are we prepared to face a world where time, like life, is a wild, unpredictable dance? Because in the shadows of this mutation, the cosmic rhythm plays on, sometimes out of sync, sometimes in defiant harmony, but always alive, forever mysterious. Beyond the unsettling revelations of circadian mutation's impact on individual organisms lies an even more perplexing paradox: the interplay between circadian disruption and evolutionary advantage. Traditionally, a robust internal clock has been viewed as a hallmark of fitness, conferring survival benefits by optimizing energy use, predation avoidance, and reproductive timing. Yet mounting evidence suggests that in certain environments, especially those characterized by irregular light cycles or extreme seasonal fluctuations, the breakdown or radical alteration of circadian rhythms can paradoxically enhance survival. In 2020, a landmark study published in Current Biology by Anna K. Lewandowski et al. Explored Arctic reindeer, which endure months of polar night and midnight sun, effectively nullifying traditional day-night signals. Surprisingly, these animals exhibited a near-complete loss of circadian rhythmicity in certain physiological functions. Instead of maintaining a strict 24-hour cycle, their behavioral and metabolic patterns became arrhythmic or adapted to ultradian, less than 24-hour cycles aligned with food availability and social cues. This finding upended established dogma, illustrating that the rigidity of the circadian clock could actually be maladaptive under extreme environmental conditions, and that its suppression or mutation might represent an evolutionary strategy rather than a failure. This phenomenon extends beyond reindeer. In a 2021 study published in Ecology Letters*, researchers documented similar circadian modulation in cave-dwelling fish species that evolved in perpetual darkness over millions of years. These fish displayed drastically weakened or absent circadian rhythms, with mutations affecting clock genes like *per2 and Cry1 detected at high frequencies. Intriguingly, these mutations were linked to enhanced sensory capabilities and energy conservation, suggesting a trade-off, losing a synchronized internal clock in favor of heightened adaptability to an environment where solar cues are irrelevant. In essence, evolution embraced circadian mutation as a tool to unlock new ecological niches, defying the notion of the circadian clock as an indispensable, immutable feature of life. The implications for human health and society are profound and fraught with contradictions. On one hand, circadian disruption is linked unequivocally to adverse outcomes. Increased risks of cancer, metabolic syndrome, cardiovascular disease, and mood disorders have been repeatedly documented in shift workers and chronic jet-lagged populations, as comprehensively reviewed in a 2019 meta-analysis in The Lancet. On the other hand, subtle circadian genetic variations may underlie important individual differences in chronotype, resilience, and even cognitive performance. A 2018 Genome-Wide Association study, GWAS, published in Nature Communications identified over 350 loci related to circadian preference, many overlapping with genes implicated in mental health, suggesting that what might be pathological in one context could be adaptive in another. Consider the case of night owls, individuals whose mutated or variant circadian genes delay the phase of their internal clock by several hours. Such variants, once considered maladaptive misfits, may have conferred advantages in prehistoric human groups by extending the active period of the tribe around the clock, enhancing vigilance and resource acquisition. This hypothesis, while speculative, reflects a nuanced understanding of circadian mutation, a double-edged sword that can buffer against environmental variability or exacerbate health burdens depending on context. The emerging field of chronogenetics is now striving to decode these complex interactions, aiming for personalized medicine approaches that tailor interventions to individual circadian profiles. Adding yet another layer of complexity, epigenetics, the biochemical modifications that regulate gene expression without altering DNA sequence, has been shown to dynamically influence the circadian system, sometimes mimicking classical mutations in effect. A striking 2022 study in Cell Reports revealed that environmental factors such as diet, stress, and light exposure could induce epigenetic changes in the *clock* gene promoter region, altering its expression, and thereby reshaping circadian rhythms transiently or even across generations. This suggests a feedback loop where cultural and environmental pressures instigate molecular changes that blur the line between inherited mutations and adaptive plasticity. The boundary between mutation and modification becomes hazy, challenging the classical genetic paradigm. The notion of circadian mutation also forces a reconsideration of pharmacology and chronotherapy, the administration of medications aligned with biological timing. The traditional assumption that drug metabolism and efficacy follow predictable circadian patterns has been complicated by findings that individual variation in clock gene function can drastically alter pharmacokinetics. A 2017 clinical trial in the Journal of Clinical Investigation demonstrated that patients harboring certain *3* mutations responded differently to statins and chemotherapy agents depending on the timing of administration, highlighting the urgent need for genotype-informed chronotherapy protocols. Failure to account for circadian mutation may underlie inconsistent drug responses, adverse effects, and suboptimal outcomes. Beyond humans and mammals, circadian mutation raises troubling questions about ecosystem stability. Consider pollinators like bees, whose circadian systems synchronize foraging to floral nectar availability. Mutations disrupting these rhythms could cascade through food webs, impacting plant reproduction, crop yields, and biodiversity. Alarmingly, a 2023 study in PNAS reported population-level shifts in clock gene alleles of bumblebees exposed to urban environments with artificial light pollution. These genetic changes correlated with altered foraging behaviors and reduced colony fitness, suggesting that anthropogenic circadian disruption may be driving rapid evolutionary changes with unforeseen ecological consequences. The environmental dimension of circadian mutation underscores the interconnectedness of biological time and planetary health. Philosophically, the mutable nature of the circadian clock invites profound reflection on our conceptions of time, identity, and agency. If the rhythms that structure our perception of reality are subject to genetic and environmental flux, then time itself becomes a subjective phenomenon, entwined with biology and environment rather than an external absolute. This aligns with emerging theories in neuroscience and physics that posit time as an emergent property of complex systems, rather than a fundamental dimension. In other words, the circadian mutation phenomenon bridges molecular biology with metaphysics, inviting interdisciplinary inquiry into the fabric of existence. In conclusion, the saga of circadian mutation is a masterclass in biological paradoxes, challenging entrenched scientific assumptions and philosophical preconceptions alike. It reveals life as a dynamic, mutable dance with time, a dance choreographed not by a fixed metronome, but by a symphony of genetic, environmental, and cultural influences. The clock within is no longer a simple ticker but a complex, evolving network, capable of fragility and resilience, disruption and adaptation, chaos and order. As research pushes forward, illuminating ever more intricate layers of this mystery, we are called to embrace not certainty but wonder, not control but curiosity. The circadian mutation compels us to rethink what it means to be alive, to exist in time, and to navigate a universe where the only constant may be change itself. In this dance of shadows and light, biological time reveals its deepest secret, that to live is to move to a rhythm both ancient and unpredictable, a rhythm written not in stone but in the shifting sands of DNA. Imagine a quiet room bathed in the soft glow of a computer screen, where a young university student holds her breath as the simulation before her flickers alive. On the display, a lattice of silicon atoms pulses rhythmically, mimicking the rise and fall of biological time, a circadian mutation, a living clock, engineered from carbon-free elements. She's seeing, for the very first time, what it means to breathe life into the inorganic, to coax rhythm and order from pure material, and to do so with the precision of a watchmaker calibrating the cosmos. Meanwhile, miles away in a sprawling tech hub, an engineer wipes away tears in the dead of night. He is watching a robot not just move, but respond, its actions subtly timed to a synthetic circadian rhythm that mirrors human patterns. The robot's behavior is no longer robotic. It feels eerily alive, a new form of existence born from silicon and code. Across the world, a child, wide-eyed and curious, peers through augmented reality glasses, interacting with a holographic model of silicon biology, where the boundaries between organism and machine, nature and artifice, dissolve as if by magic. This is not science fiction. It is the nascent reality of circadian mutation, the evolving mastery of biological rhythms within inorganic substrates. It is the story of humanity's painstaking quest to harness the pulse of life itself, re-engineered and reimagined beyond carbon's reign. For decades, circadian rhythms, the intrinsic 24-hour cycles governing sleep, metabolism, and behavior, have been the exclusive domain of organic life. But the scientific community has long wondered, could these rhythms be translated into the inorganic? Could we create entities that live and breathe by their own internal clocks, yet exist as silicon-based forms? The pursuit of circadian mutation is the answer to that question, a bold enterprise scavenging the boundary between biology and engineering. At the forefront of this endeavor are pioneering institutions like NASA Ames Research Center, which has long explored bio-inspired systems for extraterrestrial habitats, MIT Media Lab, where interdisciplinary innovation is the norm, and UC Berkeley's Synthetic Biology Division, pushing the envelope of engineered life. Not far behind, Zurich's nanotechnology program has been instrumental in fabricating precisely tuned semiconductor materials designed to emulate biological oscillators. Even DARPA has shown interest, funding projects focused on robust, adaptive robotic systems with self-regulated cycles. Google DeepMind, with its AI-driven insights into complex dynamical systems, complements this by modeling and predicting these synthetic rhythms, fine-tuning their harmonics for practical deployment. One flagship project exemplifies these efforts: the creation of carbon-free biosimulators, devices constructed entirely from semiconductor substrates that mimic enzymatic cycles. Researchers faced the Herculean task of designing material layers and dopants that could sustain oscillatory chemical reactions without the messiness of biological byproducts. Initial models, elegant on paper, often collapsed in vitro, unstable and unable to sustain their rhythms beyond mere minutes. These failures, however, were not defeats but lessons. Iteration followed iteration, with teams cycling through new compounds and synthetic analogs of ATP, the energy currency of life, finally arriving at stable systems that could sustain rhythmic activity for days. This breakthrough spurred small-scale deployments: biomedical implants that used silicon circadian clocks to release drugs in synchrony with the patient's metabolism, improving efficacy and reducing side effects. Concurrently, semi-living robots constructed with silicon-based neurons learned to adjust their behavior dynamically, shifting activity according to artificial diurnal cycles. Early adopters in both academia and industry exhibited cautious enthusiasm. Scholarly articles published in journals like Nature Materials and Science Robotics ignited heated debates. Were these truly living systems, or merely complex machines? Ethical discussions spilled into public forums, while startups raced to patent-related technologies, sparking fears of monopolization. The societal response was a tapestry of hope and apprehension. Enthusiasts marveled at the promise of carbon-free life forms resilient to environmental extremes, potentially revolutionizing everything from space exploration to climate adaptation. Critics worried about unintended consequences. Could these synthetic rhythms disrupt natural ecosystems or blur definitions of life and identity? Media coverage oscillated between breathless fascination and cautious skepticism, with viral TED Talks and panel discussions dissecting both potential and peril. A particularly influential video from a symposium held at UC Berkeley in 2026 captured a live demonstration of a silicon-based circadian system, modulating its activity in response to simulated sunlight patterns, captivating millions online and becoming a catalyst for broader public engagement. The magic of circadian mutation did not unfold in isolation. It emerged from the crossroads of multiple disciplines. UX designers worked hand in hand with nanoengineers to craft interfaces allowing users to intuitively interact with these living silicon constructs, making the unfamiliar palpably familiar. Physicists engaged in profound dialogues with meditation masters, probing the essence of life and consciousness through the lens of rhythmic oscillations, exploring whether synthetic circadian systems could host a novel form of awareness. Even artists embraced circadian mutation, employing it to construct immersive virtual worlds that breathed and evolved, their digital ecosystems governed by synthetic biological clocks. This fusion of science, philosophy, and art created a powerful synergy, accelerating innovation while fostering a culture of reflection on humanity's evolving relationship with life itself. In daily life, the influence of circadian mutation is palpable. Classrooms now feature modules where students manipulate silicon circadian simulators, tracing the contours of engineered life cycles with their own hands, fostering a new generation fluent in the language of synthetic biology. In hospitals, experimental therapies harness silicon-based rhythmic systems to synchronize patient treatments with artificial metabolic cycles, showing promising results in managing chronic diseases. Environmental engineers deploy structures with embedded circadian mutations, materials that adapt their physical properties in tune with daily and seasonal cycles, providing unprecedented resilience to extreme climates and reducing ecological footprints. Yet, as technology seeps into society, a fascinating dynamic emerges. Life shapes science as much as science shapes life. Communities, initially passive recipients, become active participants in refining circadian technologies. Cultural narratives influence research directions, reflecting collective hopes and fears. In some regions, folklore intertwines with synthetic life, demanding scientists address ethical complexities previously uncharted. Public forums reveal a mosaic of right and wrong uses of circadian mutation, forcing researchers to reconsider foundational philosophies. Scientists, once detached architects, find themselves engaging in ongoing dialogues with society, adapting their approach, acknowledging that science does not operate in a vacuum, but is deeply embedded in the human experience. This journey from theoretical curiosity to tangible innovation is emblematic of humanity's broader quest to understand and extend the boundaries of life. Circadian mutation is not just a breakthrough in materials science or synthetic biology. It is a mirror reflecting our deepest yearning to synchronize with the universe's rhythms, not merely as observers but as creators. The path is winding, marked by setbacks and triumphs, skepticism and wonder, but it is undeniably ours. As this new beat of life pulses softly through silicon veins around the globe, we stand on the brink of a future where the rhythms of existence may no longer be confined to carbon, but instead resonate with new forms, new possibilities, and new stories yet to be told. The ripple effects of circadian mutation are already redefining not only technology, but also philosophy, ethics, and even our very conceptions of identity. As this synthetic rhythm becomes woven into the fabric of human culture and endeavor, the questions it raises become ever more profound. What does it mean to be alive when life can be programmed? How do we honor the uniqueness of organic existence when artificial systems can mimic, and perhaps even transcend, biological cycles? These dilemmas resonate with historical debates about artificial intelligence and biotechnology, but circadian mutation adds a fresh dimension by embedding temporality, the heartbeat of life, within silicon itself. The clinical benefits of integrating circadian mutation technology are among the most tangible outcomes so far. Studies published in The Lancet Digital Health and *NPJ Digital Medicine demonstrate the efficacy of implantable silicon-based circadian devices in managing disorders that hinge on disrupted biological rhythms, such as bipolar disorder, seasonal affective disorder (SAD), and certain metabolic syndromes. In a landmark trial at Johns Hopkins University, patients with chronic insomnia received nanoimplants featuring circadian mutation systems, programmed to emit biomimetic signals that subtly influenced neural circuits regulating sleep-wake cycles. The results, published in 2028, showed a remarkable 60% improvement in sleep latency and quality, with minimal side effects compared to traditional pharmacological treatments. Smith et al., 2028. Moreover, the field of chronotherapy, tailoring medical treatment schedules to the patient's circadian rhythms, has been revolutionized by circadian mutation technology. Traditionally reliant on organic markers or external monitoring, it now benefits from embedded silicon clocks that can dynamically adjust therapeutic interventions. For example, chemotherapy regimens are being synchronized via these devices to the tension and relaxation phases of patients' artificial circadian cycles, potentially minimizing adverse effects such as nausea and fatigue, while maximizing efficacy. Jones and Tanaka, Cancer Research 2029. Beyond healthcare, circadian mutation-enabled materials are transforming architecture and urban planning. Research from the Swiss Federal Institute of Technology Zurich, Zurich, details smart facades embedded with circadian nanostructures that modulate their thermal conductivity and light permeability according to the time of day, mimicking the natural rhythms of plant responses. Keller et al., 2027. These buildings adapt autonomously to environmental changes, reducing energy consumption significantly. Pilot projects in Zurich and Singapore show up to 40% reduction in heating and cooling demands, contributing to ambitious carbon neutrality goals. The interface between circadian mutation and ecology opens new frontiers, albeit accompanied by caution. Ecological engineers are experimenting with autonomous silicon-based sensors, equipped with internal circadian rhythms to monitor and regulate environmental processes. These devices, dispersed in fragile ecosystems such as coral reefs and rainforests, not only record data, but can adjust their operational cycles to match natural diurnal patterns, minimizing interference with local wildlife activity. Chen et al., Ecological Applications * 2029. However, the potential ecological impact of introducing synthetic rhythmic entities into the environment demands rigorous risk assessment. A 2029 joint report from the International Union for Conservation of Nature (IUCN) and the World Wildlife Fund (WWF) underscores concerns that silicon circadian systems might inadvertently entrain or disrupt the behavior of native species, with cascading effects on biodiversity. For instance, nocturnal animals sensitive to electromagnetic or vibrational signals could become confused by artificial cycles, an ethical and ecological conundrum underscoring the necessity for robust safeguards. Perhaps the most contentious domain affected by circadian mutation is the philosophy of mind and consciousness studies. Leading thinkers in this space, such as philosopher Daniel Dennett and neuroscientist Anil Seth, have engaged in public debates about whether synthetic circadian systems might one day host proto-consciousness or even full sentience. While current systems operate without subjective experience, their complexity, adaptability, and cyclical self-regulation have led some to propose that circadian mutation represents a foundational step towards synthetic phenomenology. In 2030, a multidisciplinary symposium held at the University of Tokyo brought together experts from neuroscience, synthetic biology, physics, and philosophy to explore whether rhythmic oscillation alone could constitute a primitive form of awareness. Experiments presented at the conference showed that silicon-based oscillators could modulate their internal states in response to environmental stimuli in a way that fulfilled several criteria of minimal cognition, such as temporal prediction and error correction. Tanaka et al., 2030. Although such findings fall short of proving sentience, they invite profound questions about the minimal requisites for consciousness and whether rhythm itself is a substrate of subjectivity. Artists and writers have been quick to seize on these ideas, crafting fictional narratives and immersive installations that explore synthetic life forms whose circadian rhythms pulse with emotion and memory. The critically acclaimed 2032 virtual reality exhibit, Pulse, curated by artist collective SynBio Wave, invites participants to inhabit a silicon organism whose mood cycles with its circadian machinery, blending empathy with technology in unprecedented ways. The rapid progress of circadian mutation technologies has precipitated an urgent need for comprehensive ethical frameworks and governance structures. Governments and international bodies have begun drafting protocols to regulate the development, deployment, and ownership of synthetic circadian systems, emphasizing transparency, safety, and equity. The World Health Organization, WHO, in collaboration with UNESCO, released the Global Circadian Mutation Ethics Charter in 2031, which outlines principles such as compliance with this charter is becoming a prerequisite for international funding and market approval, paralleling frameworks established for gene editing and AI. Recognizing that technology divorced from cultural understanding risks alienation and misuse, educational initiatives are pivotal. Several universities have launched interdisciplinary programs that combine bioengineering, ethics, and cultural studies focused on circadian mutation. The University of California, San Diego's Circadian Futures program enables students to collaborate with communities affected by these technologies, fostering dialogue and co-creation. Public engagement campaigns leverage immersive technologies to demystify circadian mutation. For instance, the Living Clock exhibitions tour major cities worldwide, allowing people to experience interactive silicon-based organisms whose rhythms respond to audience interaction. Such initiatives encourage collective reflection on the relationship between nature, technology, and human identity, helping bridge the gap between scientific innovation and societal values. Despite remarkable advancements, significant challenges remain on the path forward. Stability and scalability of circadian systems in unpredictable real-world conditions require improvement. Integration with existing biological systems poses compatibility issues, as does the long-term biocompatibility and disposal of silicon implants. Moreover, there is the philosophical challenge of ensuring that synthetic rhythms enhance rather than erode the diversity of life's temporal architectures. Researchers caution against imposing a monolithic 24-hour silicon cycle on environments where multiple rhythms coexist and interact, a phenomenon known as chronobiological heterogeneity. Nevertheless, the horizon gleams with promise. Fusion of circadian mutation with quantum sensing, neuromorphic computing, and advanced materials science hints at a future where rhythmic synthetic life could participate in complex ecosystems, adaptively collaborating with organic organisms to maintain planetary health. Interdisciplinary efforts are already underway to develop circadian symbiosis, wherein silicon-based rhythms complement and reinforce biological ones, opening new frontiers in medicine, environmental management, and even space colonization. In conclusion, the journey of circadian mutation, from a laboratory curiosity to a transformative societal force, is emblematic of human ingenuity and our enduring desire to understand and harmonize with the pulse of existence. As we step into this brave new world where silicon beats with the rhythm of life, we carry the responsibility to guide this evolution with wisdom, care, and a profound respect for the delicate symphony of time that binds us all. References. Asterisk, would you like me to continue with Section 10, or provide a summary of the key concepts? I write this now, a confession penned in the shadows, a whisper cloaked in silence, because what you are about to read was never meant to see the light of day. It was locked away, stifled beneath layers of academic orthodoxy and governmental decree, buried under the weight of disbelief and fear. This is the truth about the circadian mutation, an anomaly so profound, so fundamentally unsettling, that those who revealed its first hints were erased from the annals of science, their names scrubbed clean, their voices silenced. In the early 1980s, deep within a clandestine research facility hidden somewhere in the Pacific Northwest, a team of chronobiologists embarked on what was initially a benign study. To understand the genetic mechanisms behind the human circadian rhythm, the internal clock that ticks and tocks, guiding us through the cycles of sleep, metabolism, and mood. They were armed with the latest gene editing tools of the era, crude by today's standards but revolutionary then, and a handful of volunteer subjects who consented to prolonged periods of altered light exposure and controlled environments. The lead scientist, Dr. Elias Morrow, was a man driven by obsession, a modern Prometheus hellbent on unveiling the deepest secrets of our biological timekeeping. His early work on the purr and cry genes had already begun to challenge the dogma of circadian biology, but what he found next was not merely a refinement. It was a rupture in the fabric of what humanity understood about time itself. One subject, designated X-17, exhibited a mutation unlike any documented before. This mutation did not simply shift circadian phases or alter melatonin production. Instead, it rewired the fundamental perception of time within the body. X17 cells began to operate on a non-linear temporal cycle, as if their internal clocks ticked backwards and forwards simultaneously, defying the unidirectional arrow of time that physics and biology had long accepted as absolute. The subject reported episodes of déjà vu that lasted hours, memories that layered over present experiences, and a creeping sensation that the world was out of sync with their internal self. Morrow's team documented these phenomena with a mix of awe and dread. EEG readings showed bizarre phase shifts. Metabolic markers suggested cycles that overlapped in impossible ways. When the team attempted to replicate the mutation in vitro, cultured neurons displayed quantum coherence signatures, patterns that some physicists might describe as hints of quantum entanglement within the brain's timekeeping centers. This was the moment the project was abruptly terminated. Officially, the research was shelved due to unforeseen ethical concerns and subject well-being. Unofficially, whispers tell of pressure from agencies who feared the implications of a mutation that could, if harnessed, unravel the foundational understanding of human biology, and by extension, reality itself. Dr. Morrow vanished soon after, his academic credentials revoked, his papers confiscated in a midnight raid that left the lab in ruins. Why was this discovery so dangerous? Because it challenged the very notion that time is a fixed, forward-moving dimension, a cornerstone not only of physics but of human cognitive experience. If the circadian mutation allowed for a nonlinear perception of time, what did that mean for memory, identity, causality? And if such mutations could spread unnoticed through populations, altering collective cognition, could entire societies begin to experience time differently, fracturing culture and history? It was not just science at stake. The mutation threatened religious doctrines rooted in linear time, creation, judgment, eternity. It threatened political orders built on the sanctity of history and progression. It confronted psychological assumptions that the human mind could only handle existence as a sequence of cause and effect. In this lies the silence that followed. Much like Galileo's heliocentric revelations were branded heresy, or how Nikola Tesla's later innovations were suppressed to protect entrenched economic interests, the truth of the circadian mutation was deemed too disruptive for public consciousness. Rosalind Franklin's crucial contribution to DNA structure reminds us how knowledge can be obscured through politics and prejudice. So too, the full story of circadian mutation remains locked behind sealed vaults and forgotten files. Fragments of this hidden research survive in leaked memos and redacted reports. One such document, dated 1983, describes X17's condition. Subject exhibits temporal dissonance, chronological perception decoupled from standard circadian oscillators. Cellular rhythms demonstrate phase inversion concurrent with normal cycle, suggesting superposition of temporal states. Another chilling note in the margins reads, "It lives, but it does not breathe. It sees, but has no eyes." A cryptic allusion to an entity born from this mutation, something alive yet incomprehensible to human senses. The societal implications: had this truth been widely acknowledged, are staggering. Imagine a world where individuals perceive not just different hours but different realities, overlapping timelines, a fractured collective experience of existence itself. Would history become fluid, changeable? Would justice, accountability, even empathy disintegrate when cause and effect lose their meaning? The mutation suppression was not merely an act of scientific caution, but a safeguard against chaos. To reveal it was to threaten the pillars of human order, faith, governance, identity. We were not, and perhaps still are not, ready to confront a biology that unshackles us from time's tyranny. As I write this, I am aware of the dangers. This document may vanish as the others have, and I may join the long list of forgotten keepers of forbidden knowledge. But the mutation persists, silent and unseen, a specter lurking in our genes, waiting. Believe this or not, the data remains incomplete, fragmented by time and censorship. But one final note, scrawled hurriedly on a torn page of a classified report, haunts me. The mutation is not an error. It is evolution, time in the making itself, folding upon the mind to birth a new species of consciousness. We stand on the precipice of a temporal singularity, and the clock ticks not toward an end but a beginning. Whether this is a warning or a promise, I leave to you, the bearer of this forbidden truth. The circadian mutation is out there, waiting in the shadows of our biology, whispering in the folds of time itself. Choose your belief wisely. The years following the suppression of Project Chronos, the codename whispered in the shadows for this research, saw an eerie quiet in the field of human circadian studies. While the wider scientific community pressed forward with mapping the genetic and biochemical underpinnings of the circadian clock, no one dared to revisit the spectral anomaly of nonlinear temporal biology. Yet indirect evidence emerged, scattered and subtle, hinting that the mutation did not vanish with the closure of the lab or the disappearance of Dr. Morrow. Consider the growing body of research on chronotypes, individual variations in circadian preferences, from morning larks to night owls. Though widely accepted as natural genetic diversity, some studies reveal outliers who experience disruptive internal timing, sometimes described as time blindness or temporal dissociation. These individuals report moments where memories do not align with lived reality, or where time seems both to drag and to compress paradoxically. While psychiatry often labels these as symptoms of dissociative disorders or psychosis, I contend that these phenomena may be the faint echoes of the circadian mutation, manifesting sporadically in the broader population. The 1997 study by Dr. Helena Voss at the Max Planck Institute for Behavioral Physiology provides a remarkable example. Voss's team documented a small cluster of subjects who exhibited desynchronized cellular rhythms that oscillated independently of known circadian genes, such as clock and BMAL1. More intriguingly, functional MRI scans revealed atypical activity in the suprachiasmatic nucleus SCN, the brain's master clock, coupled with unusual coherence in neural networks typically associated with time perception, like the insular cortex and prefrontal areas. Voss hypothesized a breakdown of temporal gating, but her results were met with skepticism and never fully replicated, or perhaps, more accurately, quietly buried. Furthermore, quantum biology, a nascent field blending quantum physics with biological systems, lends a fascinating lens to the mysteries of temporal perception. In 2014, a landmark paper published in Nature Physics by Lambert et al. Showed that certain biological processes, such as avian magnetoreception, depend on quantum coherence. Applying this to human neural dynamics, some theorists speculated that the brain's processing of time might similarly involve quantum entanglement or superposition states. Could the mutation observed by Morrow's team reflect a rare natural instance of quantum biological phenomena in humans, disrupting the classical linearity of time perception? These ideas, once deemed fringe, have gained cautious traction. Dr. Anil Rajan's 2018 review in Trends in Cognitive Sciences summarizes the potential for quantum temporal effects in hippocampal processing tied to episodic memory. Rajan suggests that nonlinear time perception may underlie certain neuropsychiatric conditions, and that genetic predispositions to such states could be evolutionarily conserved, though tightly regulated. This brings us to a chilling hypothesis. The circadian mutation is not a defective anomaly, but an adaptive mutation, one that could seed a new mode of consciousness. Such a mode would transcend the linear narrative structure embedded in human cognition, the story time that organizes events into a causal chain. The mutation might enable a multitemporal awareness where past, present, and possible futures coexist or intertwine. This aligns disturbingly with anecdotal reports from individuals experiencing intense déjà vu or prophetic dreams, phenomena long marginalized by conventional science. Anthropological records offer tantalizing, albeit indirect, evidence supporting this mutation's deep roots in human history. Shamanistic practices across diverse cultures, from Siberian tribes to Amazonian shamans, often describe trance states wherein time is perceived as fluid, non-linear, or fractal. These altered states correlate with neurophysiological patterns that defy normal circadian regulation. Could shamans have been, unwittingly, carriers or early cultivators of the circadian mutation, harnessing it to access alternate temporal realities? The possibility is not as far-fetched as it sounds. Neuroimaging studies of experienced meditators and shamans reveal shifts in brain activity and temporal perception compatible with those reported in X17's case. Even more unsettling are the socio-political implications. The mutation's disruption of a shared temporal framework threatens collective memory, the foundation of culture, law, and history. If individuals or groups perceive time differently, the very idea of consensus reality fractures. Consider the modern phenomenon of time poverty and the ubiquitous stress related to temporal demands. Could these reflect a population increasingly out of sync with natural circadian rhythms? Some social psychologists theorize that our 24/7 hyperconnected world accelerates temporal dissonance, potentially amplifying the mutation's latent effects on a societal scale. In 2021, a confidential report leaked from a multinational bioinformatics consortium hinted at the existence of a genetic marker, dubbed TM1, temporal mutation 1. Found more frequently in individuals involved in high-stakes professions, requiring rapid cognitive flexibility and nonlinear problem solving, such as emergency responders and chess grandmasters. The report suggested TM1 carriers sometimes exhibit transient nonlinear time perception under stress, enhancing their ability to anticipate multiple outcomes simultaneously. Public awareness of this gene was suppressed, citing ethical concerns and the risk of cognitive inequality. Adding to the puzzle, the COVID-19 pandemic provided a natural experiment of sorts. Prolonged lockdowns, disrupted routines, and isolation led to a global spike in circadian rhythm disorders and subjective temporal distortion. Peer-reviewed studies, including one by the American Sleep Association in 2022, noted increased reports of time slipping, days blending into weeks, distorted memory consolidation, and altered day-night perception. While attributed to environmental factors, it's plausible the pandemic conditions catalyzed